Good morning, everyone, and welcome back to day 4 of the workshop on finding cancer drug targets to facilitate precision and personalized medicine. We look forward to another engaging and productive day of learning together. Today's program begins with a lecture by our host faculty, Dr. Mohammad Aziz, on identification of novel therapeutic targets in colorectal cancer.
All right. Thank you, Swati. Good morning, everyone, and Assalamu alaikum. So today we are going to go further from what we have learned so far and dive into the actual uh, problem of finding the therapeutic targets. So I think we have already covered what is multiomics. So I hope all the participants here would be having now the concept of multiomics clear, what it is. Then I also covered how do we do it. And yesterday tutorial showed you some computational tools by which you can do multiomics. You can test your gene, you can test different databases, depth map, UCSC Xeno browser, the protein atlas, and you also have a wealth of data in the TCGA, the Cancer Genome Atlas. And then we have an AACR also maintains a database. So there are a lot of data that is available out there, but we need to work on it and make out sense so that we can identify novel therapeutic targets. Now, remember here, we are not looking for something very different, but we are trying to find out the targets that are for a particular patient. So that is the motive. It's not about finding new gene every time, new mutation every time. Rather, it's for finding the right gene for the right person or right target for the right person. Okay, so I will move my slides by setting the stage with the problem of colorectal cancer to be specific. So what you see on your screen is about the incidence in blue and mortality in red in from all over the world. This is a slide for the global numbers. And you can see that there are roughly 1.3 million cases in 2022. From all ages, from all regions, 1.3 million people that are diagnosed with colorectal cancer. And roughly 40% of them, they die, right? So this is still a problem, although this is a better statistics, because now due to preventive strategies, surveillance, and screening procedures, some countries like United States and New Zealand, they have reported a decline in the mortality. But when you take it globally, it's rising up in India, it's rising up in other countries of Africa. So this is still a major global health problem. Let's talk about India. So we have to have something specific. As I told you in my earlier slides, we can't generalize it all over the world. Whatever works in other countries may not work for us and vice versa. So I was looking into the numbers for India. Here also, although the numbers are low, but the survival rate is uh, pretty bad. It's just 50%. 50% of people who are diagnosed, they die. So this is all stages, all age groups, and all genders as well. So the magnitude of the problem is quite big, and this has been. I'll show you the problem that is coming up with the survival rates. So far, we have got these strategies for treating cancer: the primary surgery, then chemotherapy, the drugs that we have talked about, which are working in a systemic manner that goes in the entire body and try to target the replicating cells, the dividing cells, and kill them so that the tumor is also eliminated accordingly. Then radiation therapy is used in some cases to shrink the tumor. Sometimes the tumor is pretty big, and there is a need to shrink the tumor, and for that, radiation therapy may be used. When the options of systemic therapy, chemotherapy are not working, and the stages are advanced, then there is targeted therapy comes into play. I don't understand why they don't use it to begin with. Probably it's about the cost. It's about I don't know the reasons why they don't start with targeted therapy, but this is what it is. The targeted therapy is given to those patients who have advanced and who have not responded to chemotherapy. And the latest on the block is immunotherapy, that there are ways to boost the body's immune system, and by using the checkpoint inhibitors like CTLA or PD-1, PD-L1 inhibitors that can be utilized for curing. But this is also in the advanced stages. So we have got all these modalities available for treating the colorectal cancer patients. But still, if you look at the survival rate, so I'll walk you through the slide. These are the stages, which is called as Duke's staging system in colorectal cancer. It's in the alpha numeric format: A, B, B1, C, B1, B2, C, and D. Then we have got the tumor, nodes involved, or metastasis. This is called TNM classification. So if there's a tumor, which will be there, and if there are nodes, which are lymph nodes, are found swollen in the body, we call it as N. And if there are metastases, which means it has already spread, then we mark it as N. So if you see here, T1 means there is a tumor, but there's no node involvement. There is no involvement of there's no metastasis. So this is termed as stage 1. And pathologically, I'll show you in the next slide what does this mean. That cancer is limited to the mucosa, which is on the intestine, which is on the surface, and survival rate is pretty high. Greater than 90% people, they live, and the surgery is the main option here. Once the tumor grows in size, it's T2, but still there's no node involvement, no metastasis, but the cancer has not started going into the layers of the muscle. The chances of survival is dropping. As you can see, the chances of survival is dropping, and it is drastically low in case of the advanced final stages of stage 4. And you can see here the tumor size is growing, and then you have got the node involvement, and then you have got finally the metastasis as here as well, which is stage 4. Right. So these are the survival rates, which again remind us of the magnitude of the problem, that we need to take care of these patients, despite of having all these treatment modalities available. So this is another slide showing the same numbers in a more graphical way. So here is the start of the colorectal cancer, which is benign so far, which is on the surface. This is the surface, and this is the building of the tumor, the hyperproliferation. Then we have got the adenomatous polyps. Then there's a precancerous polyps, and then it's a full-blown adenocarcinoma. But once it starts going into the muscle, the underlying muscle, this is where the problem is severe, because the next step is the metastasis. And this is the region where it's not malignant, means cancerous. Benign means it may stay there forever without doing any problem. But malignant means it is now going to spread. And then these are the survival rates, as you just saw in the previous slide. So as we advance into the cancer stage, the chances of survival are low. This tells us about two things. Number one, the targeted therapy may be started early on. As I said, I don't know how these clinical decisions are being done. Is it driven by the cost of the drugs or whatever? And second thing is, if we are able to detect this disease early on, then the chances are very, very good. So we can develop both the screening and preventive models and preventive strategies by the help of the biomarkers. And we can also work on therapeutic targets that can help us in developing early therapeutic targets for early intervention. Now, let us see what are already known to us. And still, the statistics is so poor that people are still dying. So this is the pathway that is very much related to the colorectal cancer, the EGFR pathway, which means there is a cell surface receptors, epidermal growth factor receptor, and this is driving the rapid proliferation of the cells. So if we are able to block it, usually this is the ligand, epidermal growth factor, which if binds to this receptor, the cell starts to grow. So if somehow we are able to block this, we hope that the cells will stop to grow and the tumor will be cured. For that, there are two antibodies, cetuximab and panitumumab. Both are having the same target, but one of them is humanized, which means it has got one chain. Antibodies have got two chains, right? If you remember, the the Y-shaped structure. So the one arm is there from the humanized, and the other one is the mouse one. Cetuximab is entirely derived from the mouse. And then there are downstream pathways. So we already have these drugs which are given, but still there is not much improvement. Then we have got another pathway, which is vascular endothelial growth factor pathway. So this helps in the building of new blood vessels. So there are three types of receptors here: VEGF receptor 1, vascular endothelial growth factor 2, receptor 3. So they have developed antibodies to block these as well. So there are different drugs which are targeting different formats. Like this is aflibercept is going to disrupt all these three ligands. Bevacizumab is also going against VEGF-A. This is the ligand here. So they stopped them from binding so that this formation of the new blood vessels is stopped, because the presumption is once you stop the formation of the new blood vessels, you can starve the tumor to death. When they will not get any new blood supply, they will not get nutrients, and then they can die. And there's another one, and regorafenib is a drug which can be given to block all these three together, these three receptors together. A wonderful drug, a wonderful target, everything known, still the statistics is pretty bad. 5% survival. Remember that. Only 5% people are able to survive in this. Okay. So next is the target HER2, human epidermal growth factor receptor. This is also being targeted by cetuximab, panitumumab through EGFR, because EGFR HER1 interaction can be disrupted to prevent the cell from binding. Trastuzumab is specifically for HER2. Then in this intracellular domains, there are different drugs that are available for blocking this entire pathway. But cancer cells are really smart. They know how to work around, and even if you block this one, they might take this pathway, or they might take another pathway, and they can still survive. So this is what a normal signaling looks like. And here, when you block the signaling, we are presuming that we block the receptors, so everything is blocked, but it does not happen in the actual scenario. Another target was NTRK gene fusions. Yesterday, Dr. Asim was telling you about gene fusions as the causative events in the DNA that is leading to cancer. So like CBR, ABL mutations in the blood cancers in the kids. And in case of colorectal cancer, we have got NTRK gene fusion. This is also there in the lung cancer. That gene fusion is like gene from NTRK gene fuses with the other gene. And this fusion, this coming together makes a lethal combination that helps the cells to grow indefinitely. In fact, it's a kind of mutation that helps in the rearrangement and the formation of these fusion genes. And it is common in colorectal cancers with microsatellite instability. I'm not going into the details of the types of colorectal cancers, so that there are different types of cancers within the colorectal. But what the message you have to get is that the targets were identified based on single omics, and then they are making things better, but not to the extent that we can have full control of it. Another example here about the microsatellite stable tumor and microsatellite unstable, or the tumors which are deficient in the DNA repair, which is mismatch repair here. If we have got the tumor which is having the mismatch, the microsatellite stable, then the tumor is not expressing a lot of new antigens. So remember here, we had a discussion that if you have got a faulty DNA repair system, is it good or bad? In this case, it is good. Because once you have got a faulty DNA repair system, the DNA is having errors, and this is leading to the production of more antigens which are new. So this is the difference that you can see here. This is a stable tumor. It has got only one epitope here, which is being recognized by the T cell, right? So the response is quite poor. But if you have got a microsatellite unstable one, more errors are being done in the DNA. The mutations are more there. So we have got more new antigens, which are being recognized by the T cell, and ultimately you see a good response. So now it is well established that you don't give this kind of a therapy of anti-PD-1 or PD-L1 antibody to the patients who are having good DNA repair system. So it is given only to those patients who have got a poor DNA repair system. A little bit of personalized medicine that you can see here. In fact, in this also, if there are KRAS mutant patients, so this is a RAS gene, if it is mutated, then it does not need all this dimerization or binding of the EGF. Rather, it works on its own, and it can drive the cancer cells to grow more and more. So if there is a KRAS mutation in a patient, they are not given the therapy of this, so that because it's useless. It's not going to work. It's already mutated, and it's already going to drive the cancer cell growth, right? So it's very important to know the patient profile before we give them any kind of drugs. So we are stepping towards that, but we are not yet close. So the question that comes out is, we have seen so many targets here, the EGFR, the HER2, the PD-1, PD-L1, so NTRK fusions. Why these targets fail to improve the therapeutic outcome? I will give you a few seconds to read this and see what is the current scenario in India. I'm not talking about the whole world. I'm talking about the country where I'm speaking from. I think I have a question here. Chewy, I'll have to take time off to do this job, but you can ask your colleagues if somebody has it, or I'll ask the Dr. Afzal if you can provide the links here in the message box. Okay, so globally, India ranks 5th in the CRC mortality with 38,000 deaths in 2022 alone. And still, if you see the 5-year relative survival of colorectal cancer, it's pretty high in case of US and other countries. So India is just second to China in case of the rectal cancers, which is an alarming situation. But still, we don't see them in the priority list when they do the funding. They don't have any priority for colorectal cancer. So are we really seeing the right target, and are we really treating the patients with the proper knowledge of what works in this particular patient? If you can remember this elephant story, and this jigsaw puzzle. If we are not knowing the complete picture, then we are really missing something and we are missing the target. And this is applicable for different patients. There will be different targets. So remember, we have to put the picture together. And this is something that I'm going to tell you with some examples today that people have done and published by using the multi-omics approach. So how to put together the full picture? That's the question. So if you remember, we talked about multi-omics, that we need to put things together from the DNA level, from RNA level, from protein level, and metabolite, as well as many other omics to create the full picture. And then we can say that, yes, this is the right target for this right person. That's the precision and personalized medicine. So this is the latest report which came out in Discover Oncology. They identified a therapeutic target in lung adenocarcinoma using Mendelian randomization and multi-omics. So people are now acknowledging this, that yes, this is the right way to do it, but still we are not there. We are still have to sharpen our tools to find out how to put the jigsaw puzzle together, because it is very, very complex. It is not as easy as we usually see. So through the multiomics analysis, they found out ICAM5 as a primary pathogenic target in lung cancer, right? And they had a significant impact on how the disease was going to progress and how it was going to respond to the drugs. And additionally, they also found KLK13 and FUT8 genes. They can be the potential pathogenic target. This was, again, a population-based study, not a longitudinal study. So, but yes, they refined the population and they subtyped it so that they can have better handle on who is going to be getting benefited from this. Another report that came out in Cell Reports Medicine, a very nice paper, a multiomics analysis assisted deep learning model. So here you see the use of machine learning and artificial intelligence, and they were able to find out a macrophage-oriented module. They did not find one target. Rather, they found this module. This is the module, which means they were CD. If you are not familiar, these are all the immune cells. They were exhausted immune cells, CD8 plus T, CD4 plus T. Then there were T regulator cells and tolerant CD8 plus T cells, and the whole R2 plus macrophages. So this was the module that they were able to find out that was the right target. So they used the deep learning models in the macrophage-oriented spatial interaction map. So the study, if anybody is interested, they can look at that. This is also from last year. It's a pretty new one. And once they targeted the full R2 plus resident macrophages, that enhances the response to chemotherapy. And now look here, the chemotherapy was given, but it was given along with this full R2 plus targeting. If you can see the survival probability was pretty high in case of this combined treatment. So the chemotherapy response predictor was designed, and then depletion was done as well as chemotherapy, and tumor was found to be regressed. Regressed means it was the size was reduced. So this is another example of using multiomics, finding out a target, and then using it alone or along with the chemotherapy. So this was used along with the chemotherapy. And I'll tell you some story about our own work, what we did in this direction. And the story starts from the patients that we took the samples from. They were all colorectal cancer patients. We took their tumor and normal tissues. We extracted DNA and RNA and protein from them. And then we started to do the study step by step. So first block, we did the genomic study. And this is the microarrays. So that's what I was telling you yesterday, that we did the microarray-based study. And we were able to find out these changes. So these are the cytobands, which means the portion of the chromosomes that were found to be affected. And these are the genes that were known to be involved in colorectal cancer. But these were the genes that we were able to find out as novel. And there's a score which tells us the confidence that yes, this event and this gene is uh, pretty robust. The possibility there's a algorithm which is agistic. It was developed by a group in Harvard. Gatti Getz is the person who did it. So it's known as genomic identification of significant targets in cancer, GISTIC. So that assigns a score. So that score helps us to give confidence about the targets that we are identifying. So we found out 11 new genes here. And out of them, we were able to zero down upon a few of them and take them further. So total 144 genes were found as significant targets. And out of them, 11 were novel genes. Now here, the approach that we took was highly personalized. We analyzed each patient separately. It was patient level analysis, not that we mixed all the tumors patient together, cancer patients together, and all the normal samples together. No. We did patient-wise analysis, and that gave us this high-resolution picture and confident targets. Then we tried to put another level of omics, transcriptomics. So we took the same samples from which the RNA was extracted in the same protocol, and we ran the exon array. Exon array is meant to measure the RNA levels. When we say exon, because exon is part of RNA and we are seeing it at a higher resolution level, but the information that we're getting is about the RNA level expression of those genes. Now, this was the result using two different algorithms, alt analyze and expression console. So this was the pattern of the gene expression. So these are the gene expression. Now you will be astonished to see that whatever was changed at the DNA level, not all of it was changed at the RNA level. Only 20 out of the 144 genes were found to be differentially expressed. So that's why whatever is we are seeing at DNA is only part of the information. It's part of the picture. It does not necessarily be the same at the RNA level. So only 20 of these genes were found to be significantly altered at the RNA level, right? And from here, we picked up a few genes for further analysis and further work. And one of them was ADB8B1. And some of these genes were very interesting that if you look at this one, ABP1 gene, this was in the region of the genomic gain mostly, which means the DNA copy number was more in the tumor. But if you see at the gene level, expression level, it was downregulated. So at the DNA level, the copy number was more, but at the RNA level, it was down. So it was a complete paradox. So we suggested that there would be some involvement of the transcription factors that is showing this phenomena. And if you read the paper, you'll be able to get the full picture of it. But there were three other genes which were showing the same scenario, like BCAS1. It was having 80% gain, which means the DNA was amplified. So usually you would expect that the DNA is amplified, more DNA means more RNA. But no, here it is downregulated. The more DNA, less RNA. So this is not a linear relationship. I think Dr. Asim also mentioned that similarly from RNA to protein is not a linear relationship. And this is a relationship which needs to be understood through mechanism. So we need to know both levels, genomics as well as transcriptomics, to create the full picture, right? So now I would ask you, can you figure out which genes are altered both at DNA and RNA level? And now if you look at this, I'll go back to my slides. So look at this DNA level changes. Look at the genes within the red box. I'll give you a few seconds. If you want to take a screenshot, you can do that as well. And then look at this picture where you have got the RNA level evidence. So you have to spot out which gene or genes are common to both DNA and RNA. Anybody with the right answer can win a Swiggy coupon. I may say that. Nobody? We have got 53 people in the meeting. If you want, I can show you that again. Okay. It's a very quiet class. So if you look at this, ADP8B1 is the one that is significantly altered at the RNA level, as well as at the DNA level. This is at the DNA level changes. Very good score, 2.326. And this is also down-regulated in case of the RNA level. So it's like the elephant story. If two people are saying the same thing, you would believe them more than one person saying it, right? So here we are having two layers of evidence saying the same thing, that ADP8B1 is probably a good target. BCAS1 is there as well. And there are other MUC4, ADAM12. I'll come to that story again. So the point here to convey is that we are trying to recreate the picture using two different blocks of the evidence at the DNA level and the RNA level, and then we are more confident about the target itself. So it's ADP8B1 and there are MUC4 and ADAM12. These are all the genes that were found to be altered at DNA as well as RNA level. So that's why I'll tell you the story about the ADP8B1, which was a very probable molecule, because we also know that this is the molecule that is found on the surface of the cell. The cell surface proteins are also very good drug targets. In the DNA and RNA, we might find the target, but we may not be able to drug it. We may not be able to do anything about it. Although now the CRISPR technologies and they are allowing us to do the gene editing, but still the proteins, and especially the proteins that are available on the surface of the cell, they are the favorite among the drug discovery people. This ADP8B1, we discovered it to be associated with colorectal cancer. And since it was downregulated, we proposed that this is a tumor suppressor gene. Let me show you what we did next. So this is the data from the patients that it's showing that it is downregulated. Then we saw it in different patients individually. Yes, it was all downregulated. Then we tested it in the cell lines. Now cell lines are different. You can see only two of them are showing down, or three of them are showing downregulation. The rest of them are showing opposite, upregulation. So we needed a good model. So this was perhaps SW480 will be a good model, or GD116 will be a good model. These are all colorectal cancer cell lines. Right. Then we used the shRNA to knock down the ADP8B1 gene. Once we knock down the ADP8B1 gene, what will happen? It's a tumor suppressor gene. If you knock it down, what will happen? Sorry, Padmavati, we just moved. Can you answer this question? If we knock down the tumor suppressor gene, tumor will grow. Very good, very good. So tumor will grow, and you can see in this green line is showing better cell proliferation rates than the control. So this is showing that the tumor is growing faster and giving us a hint that yes, your gene is a tumor suppressor gene. Next, we utilize shRNA because we always use multiple evidence. Now we try to put ORF. ORF means we try to put another copy of the gene into the tumor cells. Now, what would we expect? If you overexpress a tumor suppressor gene, what would we expect? Anybody? Absolutely. Thank you, Padmavati. And Mustafa, it will reduce the tumor growth. And that's what we saw, that in this green line, the tumor cells were growing slower than the control one. This is the proliferation rate. And this is a very nice equipment. It's called Excelligence, which gives you the real-time data about the growth of the cells. But this is all inside the cells. We need to test it in the preclinical models, and I'll tell you about some preclinical models as well later on. So gives us in fact, we also use CRISPR. I have not shown you that data, but CRISPR and shRNA, they were able to increase the proliferation rate, whereas the ORF, which is open reading frame, which means we are putting another copy of the gene, which was able to suppress the growth of the cells. So this proved to some extent that this ADB81 gene is a tumor suppressor gene. But how is it working? It's still unknown. And if anybody wants to take up that as a molecular biology problem, that will be wonderful. Because ADB81 is a molecule on the surface, and the mechanism by which it is working as a tumor suppressor is still unknown. Now, we have got another piece of the puzzle, which was metabolomics. So we did not have any setup for doing the metabolomics at that time. So we collaborated. This is where the tutorial about consortium and other things would be helpful for you, that you need to collaborate. You need to go across the department and shake hands. So I contacted a person in Sweden, and he gave us the genome scale metabolic models. These are known as GEMS. So we take our transcriptomics data, and he fed it into the genome scale metabolic models and identified some targets. And we also did the PCR validation, quantitative RT-PCR validation, to find out which metabolic pathways were significantly altered in our patient samples. So this is a complex pathway, but I can tell you that this was the glutathione metabolism, which was getting affected in these patient samples. And the genes that were affecting that was ODC1, SRM, and SMS, SMOX2, and RRM2. So we collaborated, and we found that probably these are the target genes. Now, these genes were differentially altered in different patients. So again, personalized medicine would be helpful. We look for these genes in individual patients and see which one of them can be helped by targeting these genes and which one of them will not be. So here I will introduce for some early learners this technique that we use for doing the validation. I missed that yesterday. So quantitative real-time PCR, which is going to give you an idea about the copy number of mRNAs present in the sample. So we can use different chemistries. There is Stackman probe, there's molecular beacon, and there is a simple one called cybergreen. It's not here. The cybergreen is a dye that just binds to the double-stranded DNA, whereas Stackman probe and molecular beacon are very specific for your DNA. And when they bind, they also emit signals, light, which is recorded. So as you do the PCR, I'm presuming that all of you know about the classical PCR. With every cycle, you are amplifying the number of the copies of the gene. As you are amplifying, increasing the cycle number, the fluorescence intensity also increases, and it gives you an idea about the amount of RNA that was present to begin with. So if you've got less RNA to begin with, it will take more cycles to come to the threshold. If you've got more material to begin with, it will take less cycles to come up to the threshold level. So we utilized this technique, and we validated these genes. Now, if you look at, these are different patients. And in these patients, they are upregulated, while in some patients, this is downregulated. So if I'm to give a therapy which is against ODC1, in fact, there was a drug that was in clinical trial, tazarotene. It worked in some patients, did not work in some patients, and it was stopped because half of them were not treated. So here you can again appreciate the personalized medicine approach, that this patient would not be benefited with ODC1 inhibitor. Rather, this patient would be. So we need to profile the patients first, and then only we give them the therapy. Similar was the case with other genes, RRM2. This was pretty uniform, like most of them were having upregulation. Same in cases MOX2, SAT1. If there was something that was upregulated in all the patients, yes, that we can say as a master target. If you remember my previous lectures, there were quest for master target. This is what the industry looks for. They're looking for master targets. But in cancer, I can tell you this is impossible because every patient is different. And overall, this was all done in the cell lines. So this was even in cell lines, ODC was showing a very uniform behavior, but in other ones, there were different responses in terms of the expression level of these genes. And we use the Tachman chemistry for validating these genes. So we did genomics, we did transcriptomics, and we did in silico metabolomics. We were not able to do proteomics, and because I left that institute before we could finish that study. But we did combine genomics and transcriptomics for another purpose. We used this fusion of genomics and transcriptomics, and we created signatures for classifying the patients so that we can categorize the patients, and then we can give them the therapy as per their subtypes. This was in collaboration with another scientist, Professor Hautaniemi in Finland, and he's a computer scientist. He's not a biologist. He's not a clinician. He does not know anything about cancer, but he did this study, and we were able to have 3 different classes of patients with 3 different survival rates. And these were the survival statistics for these patients based on the data that we provided to them. So see, when we fuse the transcriptomics data and the genomics data, we can use it even for finding out the group of patients, and we can tell them that how are they going to progress in their disease. So one group of patients with the red line, they have got the poorest survival probability, right? And the blue one is better and the green one is the best. So if a physician has to make some decision about treating them with certain drugs, they can have some information about which one to prioritize and which one to be given an alternative therapies. Then we also use this data for creating some prediction models, support vector machine-based models. I had a student from Africa, and he developed this prediction models to tell about the cancer in normal samples. So he also had, but this was a method, computational method, where we did the feature selection, and then we used the support vector-based, machine-based prediction models. This was the story that we did for finding out the target. Am I audible still?
Yes, sir.
Okay, let me just mute this notification I'm getting. All right, sorry. Sorry about the disturbance. Somebody's trying to call and it's coming on my computer as well. So therapeutic targets can also serve as good biomarkers. So the multiomics exercise that we do, it's not just about finding targets. We can also do it for risk prediction. We can find out biomarkers. We can use it for classification. And the ultimate objective is to help the patients. So therapeutic targets can also serve as good biomarkers. So when we are looking for the genes, or we are looking for the proteins, or we are looking for the targets that are altered in tumor cells, they can also be used to tell us about the progression of the disease. They can also be used to tell us about even for the diagnosis of the disease, or for the response to the drugs. So they are known as diagnostic biomarkers, which means anything that we can measure and it can tell us whether there's a disease or not. We call them as diagnostic biomarkers. Then prognostic biomarkers are the one which are the measurable entities that can tell us how the disease will progress. Will it be fast? Will it be slow? Will it be worse? Will it be benign? And then we have got the predictive biomarkers, which can tell us that whether this particular drug will work or not. So these are known as predictive biomarkers. So from the same study, we tried to pick up some of these molecules, some of these targets for serving as biomarkers. And there was another study, before I tell you my story, this was another paper that utilized the same multi-omic data set. And yesterday I saw some interest in some people who are trying to do the meta-analysis. So I thought of putting this slide for them, that there's a lot of scope because there's a lot of data that you can utilize and do these meta-analysis and find out the targets and biomarkers. So they have utilized the TCGA, the Cancer Genome Atlas, and Gene Expression Omnibus. And then they did the statistical analysis, machine learning models. But you have to, if you validate these studies in the in vitro and in vivo conditions, they become very good reports. You can publish them in high quality journals. And then you get the biomarkers, which are diagnostic, prognostic, or therapeutic, based on different omics that they have taken up. So this is just to give you a reference for doing some of the studies that you are interested for multi-omics. Okay, coming back to the same slide that I had shown before, we have got other genes that we utilized for the biomarker analysis. So there was one that is here, MUC4, which was downregulated, and there was another one, which is ADAM12, which was upregulated. So we thought of choosing these two because these are significantly altered at the DNA level as well as the RNA level, and utilize them to see whether they are good biomarkers or not. So same approach, but for a different purpose. So we have utilized it for identifying the targets. We have utilized it for the classification of the patients. Now we are going to utilize it for identifying the biomarkers. Now we want to use it as prognostic biomarkers, which means we want to know how the disease will progress, and if these two molecules can tell us about the different stages of the disease. So for that, we needed a cell line model because you can't do these studies in patients unless you have got a very good data at the preclinical and cell line level. So we choose these three cell lines, CCD841, which represents a normal colon cell, then SW480, which represents a primary colorectal cancer, and SW620, which represents a advanced metastatic colorectal cancer. So we check the expression of these gene MUC4 and ADAM12 in these cell lines to know if there is any changes in these states. If there are any changes, yes, we can use them as biomarkers. So there was another part of the story where we did not utilize the entire gene. Rather, we tried to look at the exon level. So we looked at different transcripts. So what you're seeing here is ADAM12 all, which means it's a complete gene. But then there are different variants, 245 and 1213. So these are different transcripts of the same gene. I'm not sure how many of you are familiar with alternative splicing, but with alternative splicing, I touched upon yesterday that with alternative splicing, you get the different formats of the gene from the same pre-mRNA. So we utilized not the gene itself, rather the transcripts, that isoforms we call, for use as biomarkers. And we quantified them in different cell lines, and we tried to find out if there are any changes. Then once we have these targets or we have got these biomarkers, there has to be some preclinical models to test them, right? I've just put this single slide here, but the entire paper is available. I've given the reference here. If you are interested, you can go there. I'm trying to make it very simple as well as useful. We have a diverse audience. So all that we found here was that yes, Adam 12 and MUC4, both of them can serve as the biomarkers, the prognostic biomarkers for these for the colorectal cancer progression. Now, once we have the targets, the problem, especially for the personalized approach, that how do we test them? So there has to be either we take thousands of mouse, or we take thousands of cell lines, or we take patient-derived organoids, which are 3D structures, or there was somebody very smart, and he was a good friend of mine. Now he's relocated to Ireland, and he made some fly avatars. This is Drosophila. So what he did, he took the patient's genomic analysis. The only problem here was he did not take all DNA, RNA, protein together. Rather, he just looked at the mutations. But then he took these mutations and he made the engineered these Drosophila and made it as good as this patient. And then, since Drosophila you can culture in the Petri plates and you can culture many of them, they were able to test thousands of drugs on it. And then with that, they were able to find out which treatment is going to work and which treatment is not going to work. And then they give you 5 treatment options for that patient to be implemented by the oncologist. So this is now already a company that is being run by Laura in UK. And I told you another company, Nantomics, which is also doing multi-omics rather than just the DNA mutation profiles. So they had identified different oncogenes, a tumor suppressor for different variants. They have done it for a couple of cancers, but the final outcome for all this is to be precision and personalized medicine. And that's what we are hoping to do with the multi-omics as well as the personalized medicine approach, that we can provide the right treatment to the right patient at the right time. I think with that, I will stop here and take any questions if you have. We have got 8 minutes to go. If you want me to go through any slide again, I can do that. And if you've got any questions or discussion points, feel free to let me know. Okay, so I'm hanging around to try to absorb all of this, and I can run the slides once again so that you can have an idea. Or if you're seeing it on YouTube, you can even scroll back and see that. But for you to be able to see it.
Thank you very much, sir, for this highly informative session. We'll wait for a few more minutes for people to ask some questions, and then we
Okay.
will move on to the next session.
All right, that's fine. So I'm turning off the camera, but I'm hanging around. So if you have any question, you can speak or you can write down. Thank you. Thank you for your listening. Well, Sandeepan, these models, they were validated first that whether they actually recapitulate the human system and human mutations. So validation means first they were tested with the drugs, and then it was also tested in other animal models. And it was found that yes, this was working. So the considerations here are numbers. If you can make as many tests as possible, that is the best. So if you test only 10 drugs and tell, okay, these two will work, it will be inferior to what if you say we have tested 1,000 drugs or 5,000 drugs, and then we are giving this recommendation. I would say increase the number of drugs that you test, and then give the recommendations for which drugs are going to work and which one is not. But that is going to be expensive as well. And that's why this is unaffordable for patients in India, that it's costing about $100,000 for a patient. Okay, DNA changes do not always correlate with RNA level changes. Why is that? Okay, the first and simple answer is epigenetics. So DNA might be duplicated, but there is a silencer of methyl group there. It might not express at all. If you're seeing something duplicated at the DNA level, it would not be transcribed into the mRNA. I hope that answers your question, Mustafa. And there are other mechanisms as well. Swati, I have made Hifzer as a co-host.
Yes, sir.
Okay, good. So in 2 minutes, we'll reconvene. I think, Swati, you can utilize this time in introducing
Yes,
Dr.
sir.
Hifzer.
All right. So once again, sir, thank you very much for this highly informative session. And now it is my pleasure to introduce Dr. Hifzur Rahman Siddiqui, a senior assistant professor at Aligarh Muslim University and program director of the Molecular Cancer Genetics and Translational Research Program. He earned his PhD from the Indian Institute of Toxicology Research and completed postdoctoral training at leading universities in the USA. Dr. Siddiqui's research focuses on cancer biology, pharmacology, and chemoresistance. He has published over 75 research articles in high-impact journals and is recipient of several prestigious honors, including the SPUR Young Scientist Travel Award by USA, IABS Outstanding Cancer Research Award, and the SPER Innovative Researcher Award. We warmly welcome Dr. Siddiqui to deliver his talk on epigenetics and cancer. Thank you very much, sir, for joining us. Over to you.
Thank you, Swati. Thank you very much. And I am extremely sorry because due to the maybe there is a temporal epigenetic changes of my tone. So before going to start, where is the window? Yes. Is it visible now?
Yes, sir, it is.
Is it visible?
Yes, sir.
Okay. Thank you. Thank you very much. It is not expanding. Yes. So the topic is epigenetics and cancer. So this is a hot topic, and this is the recent topics. Most of the scientists working, the number of scientists working on this particular field. Now, before going to talk on the epigenetics part, and I am giving you means you people are listening so many things on cancer, precision medicine. So I'm just giving you a few sentence on cancer. So this is the scenario of cancer. It is now it's 18 means maybe it is around 20 millions, and into by 2040 the statistics would be almost 30 millions. Means maybe the difference is almost 12 million. So means and the number of things if you change particularly our lifestyles means we can prevent almost according to the USA American Cancer Society we can prevent almost 42% cancers. In Indian case according to the ICMR we can prevent almost 60% cancer just by changing our lifestyles. So this is the means a few line on cancer, and now the main topic epigenetics. Now the question is what is epigenetics? The Greek word epi, which means above or on the top, and that is part of the genetics. So the above or the changes which is above the genetics. Genetics means this is the we know what is the simple definition of genetic inheritance and variation, and the key molecule is the DNA. Now, the epigenetics refers to the study of how genes are turned on and turned off without altering the DNA sequences. So, without changing our DNA genetic compositions, how the gene expression is turned on or turned off, or how this gene expression is changes during our life or during the disease conditions or during our different lifestyle conditions. Now, this term epigenetics, which was coined by C.H. Waddington, you can see. So he is the scientist who coined the term epigenetics. Now you can see from the right pictures, there is a book. So if you consider the book is our genetics, the marker, this here book marker, they are the epigenetics. So the content is same. The content is same all the persons or the same persons, or maybe you can consider the for genetic purpose you can compare the twins. Okay, I will show you some pictures. You can compare the twins, monozygotic twins. They have 100% similar genetic compositions, but the bookmarks or the epigenetic marks, they are different from person to person. Even in monozygotic twins, those have the 100% genetic similarity. Now, this molecular modification of chromatin, some of these modifications are here, but we'll discuss later in details. So before going to this, I will show you a 2 to 3 minutes video. This the summary of epigenetics.
There are over 100,000 skin cells in 1 square centimeter of skin. Each of these cells contain close to 2 meters of DNA, which contain our genetic information. All the genetic material in our body is called a genome. Because our DNA is very long, cells package up their DNA together with proteins known as histones. The DNA and protein complex is known as chromatin. DNA is wrapped around histone proteins, forming repeated units of nucleosomes that look like beads on a string. Chromatin is condensed further to form a chromosome. We have 46 chromosomes in total, stored in the nucleus. The core of one nucleosome is composed of DNA wrapped around a histone octamer that consists of 2 copies of the major types of histones, H2A, H2B, H3, and H4. This organized DNA protein complex allows the cells to regulate what genes are expressed and when. All the cells in our body contain the same DNA sequence. Skin cells, muscle cells, and liver cells contain the same DNA sequence. Yet all these cells have different structures and functions. The reason is that different cells only use or express certain genes. Within each cell, the DNA and also the histones can be tagged by tiny chemicals that modify gene expression. These chemical tags cause some genes to be turned on and some genes turned off. For example, muscle cells will have genes turned on that a muscle cell requires, and will turn off genes that a liver cell requires. Similarly, the liver cell will have genes for muscles turned off and genes that a liver requires turned on. These modifications are known as epigenetic modifications. Epi is Greek for above the genes, so modifications above the genes or on the genes. Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides: G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG islet. And here the cytosine are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modification. This is where acetyl or methyl groups are directly added onto histone tails, and this will modify gene expression. For example, histone 3K9 acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modification, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
So this is just a short video, just to summarize what is epigenetics and how it's related to our DNA and histone proteins, the proteins which wrap the DNA. Now here you can see this is the monozygotic twins, and beautifully you can see, though they have the 100% genetic compositions are same, 100% monozygotic twins they have the 100% compositions are same, but you can observe their hair color is different. And why this hair color is different? This is the questions. Here you can see this is the same dog. Okay, the 2 eye color is different. So this is here it comes to the epigenetic regulations. Now, how this epigenetic in or genetic compositions as well as epigenetic influence our whole phenotypes. So these epigenetic influences can be influenced by, you can see, microbiota. The microbiota, you know, this microbiota present, the millions of microbiota present in our mouth cavity as well as in our lungs, as well as in gut. Then also these epigenetic changes are also influenced by diet. Suppose this epigenetic whole composition is, you can find from, suppose methionine rich food. If someone take methionine rich food, they have a different epigenetic composition, and then maybe they are siblings, or maybe they are twin sisters or brother. So diet also influence this one, because it means here it has been already reported that suppose those who are non-vegetarian and those who are vegetarian, they have different epigenetic compositions, as well as some means in race wise. Suppose if some Brahmins, because they are mostly dependent on vegetarian food, their epigenetic composition is quite different from the Rajput or Patan. So now this is here you can see another is a disease exposure. Some disease also, some bacterial or some viral also influence this disease epigenetic compositions or epigenome. We will discuss the details about the epigenome. What is epigenome? Then psychological state, pollutants, lifestyle, toxin, chemicals also influence your epigenetic influence, and then exercise or physical activities. So this combination of our genetic compositions as well as the epigenetic or epigenome combined together to form our whole human phenotype. So here this picture I have taken from the NIH website, and here you can see how this epigenetic mechanism starts from the uterio to the childhood to the adulthood, and from the aging up to the death. So here you can see these changes. Sometimes it is a permanent, sometimes it is a transient. Mostly it is transient. So here you can see this epigenetic mechanism. They are developmental, environmental chemicals, drug, pharmaceuticals, aging or diet, and they how they in already you have shown the whole picture in the video. So this is the histone tail, and this is the target for the epigenetic, mostly target for the epigenetic modifications other than the DNA sequences. So, as you studied during the undergraduate or postgraduate molecular biology, here you can see this. We have there are two types of chromatin in our chromosomes. One is euchromatin. So, what is the meaning of euchromatin? Eu means true. This is the true chromatin. True chromatin. Why it is called true chromatin? Because they control the gene expressions. And another part of the chromatin is heterochromatin. Means that there is a famous chromosome. You have studied the polygyny chromosomes or Lembrash chromosome. In polygyny chromosome, here you can start with the band and interband. So this interband part is the euchromatin and the band part is heterochromatin. And why it is a heterochromatin, here you can see these heterochromatin part or band part, they are highly compact. Why they are highly compact? Because of the high level of methylations. And this methylation causes the compact nature of this heterochromatin. And euchromatin, mostly here you can see this less methylations, more acetylations, and this acetylation making it accessible to different proteins, and that's why proteins like RNA pol or DNA pol, RNA polymerase or transcription factors and other things, and so they can express easily. So this is means within the chromosome you can see this is the difference between the how these epigenetic changes regulate the gene expressions in euchromatin or true chromatin, where the gene are highly expressed or they are active part of the chromosomes, and heterochromatin, they are the inactive part of the chromosomes. Now here you can see this, suppose this is a person A and person B. You can see this is the white blood cells, or you can take any cells, okay, what is it, the hepatocytes or skin cells or pancreatic beta cells, gamma cell, whatever it is. So you can take the two cells from the person A, B from the similar type. Here the example is white blood cells. Both the person have the same DNA compositions. So DNA is same, nucleotide sequence are same, their genetics are same. But when this epigenetics changes, here you can see here the epigenetic is different from two person A and person B. Here you can see 100% methylation is there, but here it is 50% methylations. Now, depending on this methylation, maybe the gene will is going to be a turned, some genes are going to be a turned on, or some will turn off. So currently, here you can see some examples. This is the Nature recent paper in Nature Cancer. So there are so many medicine targeting the epigenetic targets. Here you can see there are so many medicine already there. Some are in the clinical under clinical trials, phase 1, phase 2, or phase 3, or some are already in the clinics. Few are in the clinics. Here you can see this is the some of these drugs and how they target different epigenetic modifiers. Or there are three types actually: modifier, writer, and recognizer. So we'll discuss in later in our slides. And these are the other than the cancer in other field also you can see. So here you can see these are the suppose this blue color, they are the neurological modifier, okay, epigenetic targets of neurological diseases. So some are the in yellow, they are the viral infections, and this green color here you can see both this green color is immunological, but it is also used in cancer. So next is another beautiful examples we all have studied during our BSc classes. This is how this environment influence the epigenetic changes. Or here you can see when a mouse take the normal diet. So this is the normal diet they produce. There are so lots of yellow color plus obesity prone diseases, obesity mouse. But when this diet, normal diet, is modified with adding vitamin B12 or folic acid, choline or vitamin, you can find the lean mouse and obesity is less, and they are not prone to the disease. So I have taken this is from a book. These are the from a book. This is the citations reference. So this is also I have taken the same things here. They represent in NIEHS, National Institute of Environmental Health Science, USA. This is the examples how this environment or diet can influence your epigenetics. So now the question is how this happens. Then what is the mechanism behind this? One example, this is the case is called agouti viable yellow genes. This gene is only found in mouse, not in human. Though human and mouse both are mammals and they have a very close genetical relationship. But here you can see this particular gene only found in the mouse. This AB locus, okay, this is N transposons. This AB locus is actually retrotransposon, that is jumping gene that is inserted upstream of the agouti genes. Okay, this is the agouti mouse. They are called. This particular gene is present and they are present in a means number of copies present in the mouse genome. But when happen, these AB elements are methylated. Okay, this particular transposon is methylated and they are normally shut off. They are not turned on. They are shut off or turned off. However, when agouti mouse, they are unmethylated. When they take some diets, this methylated locus is unmethylated, and when it is unmethylated, the promoter is active, and they leading to the yellow coat. The gene express leading to the yellow coat, and they are highly obese mouse as well as they are prone to different diseases. These environmental effects as well as diet you can also observe in other parts. Here, one example is in human condition, how the psychological stress can influence. This is one example is children who were conceived during a harsh wartime famine in the Netherlands in the 1940s. Here you can see they have the more diabetes, heart disease, and other conditions in Netherlands in 1940s. And this study already published. These are all are available in the online. So it means in other conditions, you can also means if you compare means two families, means suppose one mother is under stress condition, he deliver a child, and one in normal condition, you can observe the psychological difference of the both children. So this is the summarize of this is the latest one. Means one of this beautifully I have taken. It is from the Frontier Cell and Developmental Biology, published in 2014. Here you can see these are the all things already I have discussed, shown it. Psychological stage, social interaction, your epigenetic composition is completely dependent on all these things. Even your friend circle, social interactions. If you have a bad friends and if you have a good friends, your epigenetic composition will be different from those who are interacting with the good people and those who are interacting with the bad people. They have the epigenetic composition is totally different, and these are all published data. Okay. So your social interaction is also depends alternative medicine, therapeutic drugs, microbiome, exercise or physical activities, financial status, because you are depending on financial status, maybe you are under psychological pressure or economic pressure. This is the drugs of abuse, toxic chemicals, disease exposure, diurnal seasonal correlations, etc. So now, before going to the precision part, we should know the historical background of this epigenetics. Okay, so because this is though it means complete work on epigenetics started after 1951 or 1942, but though 1942 it was discovered, but the work has been started after 1957 or 51, you can say. So in 1751, first the theory of epigenetics was given by C.F. Orr. And according to this theory, you have started this theory in evolutionary biology book. Structures that have not yet developed emerge throughout the development. Okay, throughout the development, what does it mean? It means your genetics are same. You inherited from your parents. Genetics is same, but it is developed because those organs which are pre-developed, they are developed because of the changes of your gene. And these changes is not the nucleotide composition. The changes in the epigenetic changes. But at that time, it was not discovered what is epigenetics or something fixed. So now 1802, Lemmer proposed the environment might directly or relatively alter phenotype. And you know, this is the his most famous theory, use and disuse. And this one is also one of the most famous theory by J.P. Lemmer. He was a famous French evolutionary biologist. Then in 1879, Fleming coined the term chromatin to refer to the structure of the sustainable cell nucleus. In 1898, discovery of nucleotide by Raphael, known as tuberculinic acid, which is not recognized as a prerequisite or identification of DNA methylation at that time. In 1925, Johnson and Coghill reported detecting a minor amount of methylated cytosine, and this is the first identification of epigenetic changes of a nucleotide. Now, in 1942, the term epigenetics was coined by Waddington of Cambridge University. 48 discovers first naturally modified DNA nucleotide, that is the cytosine, in a chromatography of calf thymus DNA. 51, wet first the isolation of 5-methylcytosine, then create a model of epigenetic landscape, the same Waddington who coined the term epigenetics. Then size 61, chromosome inactivation, we have by Leon hypothesis, you know it. Then 62, Arbar and Lin discovered histone methylation. 64, histone acetylation. Then 65, DNA methylation. First structural resolution of nucleosome, it was in 1974. In 1975, DNA methylation proposed as a process for the embryonic silence of X chromosomes. This idea that DNA methylation may regulate gene expression was proposed by Holliday and Ricks. 77, discovery of histone ubiquitination. First proof that DNA methylation is responsible for X chromosome silencing. So we know this. It was discovered that this bar body or X chromosome silencing means we have studied in genetics. It was discovered earlier, but first time it was proposed that or proof that DNA methylation is responsible for X chromosome silencing. Then bromodomain or prions in 82, 85 to several divide acycline or cytotoxic agent developed by Upjohn inhibits DNA methylations. Then some drugs were discovered around 1982. Then this widespread loss of DNA methylation found on cytosine guanine island in a tumor samples. And from onwards, it was discovered in John Hopkins University, USA. And 1982 onwards, this epigenetics was related to the cancer. So there are so many people started to explore the possibility of epigenetic changes and how they are related to the cancer. Not only cancer, other disease also. So this is 85. Again, DNA methylation takes place on particular DNA regions known as CPG islands. Then cloning of this DNA MT or DNA methyltransferase genes. Then suggested inactive tumor suppressor genes. DNA methylation suggested in 89. They first proposed that DNA methylation is responsible for the tumor suppressor genes. We know there are two types of genes found actively involved in cancer. One is oncogenes and another one tumor suppressor genes. So tumor suppression, they suppress the genes, and oncogenes, they promote the genes cancer. So oncogenes promote the cancer and tumor suppressors suppress the cancer. Then subsequently, there are so many discoveries. You can see the first conclusive study to show that decreased methylation of DNA resulted in the onset of cancers. And this methylation specifically, they decrease on the tumor suppressor genes. Sorry, oncogenes. Now in 1996, discovery of histone acetyltransferase and histone deacetylase, discovery of RNA interference, and first 1999 first demonstration in mammals is that epigenetic alternation may propagate through the generation from one generation to another generation, and then also linked with the colorectal cancer. In 1999, first time it was reported that. Then in 2001, chromodomain obtaining protein was discovered. Then 2004, identification of FDA approved and DNA methylation drugs first in 2004. Then 2006, another drugs. And I have shown you there are so many drugs are online. They are either in clinical trial 1, 2, 3, 4, or some are somewhere in the clinics. Now the next question is, what is epigenome? We have started the epigenetics. Now the next question is, what is epigenome? The epigenome is a multitude of chemical compounds that can tell the genome what to do. Means it includes, epigenome includes not only the methylation, it includes the acetylation, ubiquitination, somalylations, acetylations, or phosphorylation, etc. The human genome is the assembly of DNA. We know it. Almost there are approximately 3 billion base pairs are there in the genome. And this epigenome is made up of chemical compounds and proteins that are attached to DNA and direct the action of genes, whether it should be turned on or turned off, or shut on or shut off, controlling the gene expression in particular cells. Some examples are there. Suppose we have the same DNA, both in hepatocytes. islets of Langerhans or gamma cells, beta cells, skin cells, mouth cells, blood cells, WBC, RBC, every not RBC or WBC, WBCs, and other cells also. Maybe in brain cells or neuronal cells, we have the same DNA. In same person, they have the same DNA. Now, how the cells know? Means where already in the video you have seen in the liver, what happens? Or in the liver, in hepatocytes, some genes are expressing. In the muscle, some genes are expressing, but not the other genes. Suppose in insulin gene is expressing only in the pancreas, not other organ. So why? Why? What is the reason? Because they have the genes, they have the gene sequences. So these controlling or differential gene expression is largely controlled by this epigenome. Now, what makes up the epigenome and what does epigenome do? The first is what makes up the epigenome? The epigenome is the set of chemical modification to the DNA and DNA associated proteins. What is here? This DNA associated proteins, maybe the histone proteins, which alter gene expression, and they are sometimes heritable through meiosis or mitosis. Now, the modification occur as a natural process of development or tissue differentiations, can be altered in response to the, I have already told you, different reasons, maybe diet, environmental exposure, disease, or microbiota. We have a trillions of cells specialized for different function in muscles, bone, etc. Each of these cells carries the same genome. The epigenome controls many of these genes to the genome. In this way, cell can remember which genes are turned on and turned off, when, where, and how. Now, these are the summarize of the epigenetic changes we observed. This is, I have taken, this is from one of my recently publications paper. So these are the DNA methylations, histone modifications. These are the some examples. And this histone modification, then there is a microRNA or long non-coding RNA, specifically you can say non-coding RNA. They are also involved because this non-coding RNA include this here. I have mentioned only microRNA, but some non-coding RNA, they are directly involved with the, suppose, one example, very good example is X chromosome inactivation. And this X chromosome inactivation is, this chromosome is inactivated by a long non-coding RNA. This is called cyst. They wrap the whole chromosomes and the chromosome is unable to express, okay, throughout the life. Then another one is, this is the RNA methylation. This is a new field. And this recently we observed that maybe in 2010 or 2011, something like this. This N6 methylation of the adenine residue of mRNA, these are epigenetic changes in RNA, and they are directly related to the gene expression as well as in cancer biology. There are so many people are working with this, specifically some of these enzymes which involved in adenine methylations. They are MET L314, MET L14, H16. There are, means you can just make a search on PubMed, you will find there are so many papers are coming. This is a very hot field nowadays in cancer biology. Now, this major multiplication here, you can see this, I have already discussed. This is the methylation, acetylation, ubiquitinations, and this is a not a very recent data. It was published in 2016 in Nature Reviews. Here you can see this is the methylations, means in the histone, which histones are methylated where, and what is the effect of this methylation. Sometimes methylation not always repress or suppress the genes. Sometimes they activate the genes also. And here one example is H3 histone 3K4. Here you can see this is the acetylation, some of the, but another means in general, we understand that methylation causes gene suppression, acetylation causes gene expressions or activation. But this is not always true. Here you can see sometimes the acetylation also causes repressions, and this is the ubiquitination. Now, the histone molecules, we know, we already discussed the DNA. Means we have the main target is the CPG, and sometimes also in adenine, guanine also. But in the most epigenetic changes, what we observed, this is mostly in the histone tails. And these histone tails here you can see, this is the H2A, H2B, H3, and H4. We, as already you observed in your video, there are 4 tetramer of this histone proteins, and they form the nucleosomes, and this the DNA wrapped around the nucleosomes, and they compact. Finally, they form chromatin, then chromatin to the chromosomes, and chromosomes, how they are compact. We observed what we observed during the mitosis or in under the microscope. So here you can see these tails, H2A, these are the tail regions of H2A, H2B, H3, and H4. And these are the different types of modification we observed in the tail part. Here you can see phosphorylation, methylation, acetylation, or ubiquitination. Now, these modified tails, then they recognized by some domain containing proteins, and these domain again divided into the four parts. They are bromodomains, chromodomains, pseudodomains, and PSD domains. And these proteins, when bind to the modified part of the histones, what does it mean? These bromodomains containing proteins interact only the acetylated histone tails. Then chromodomain or pseudodomain proteins contains only interact with the methylated histone tails. Means histone tails, here you can see this is the, I am talking about this one. When it is a methylated or when it is an acetylated, a particular type of protein comes and bind to the tails, and where these proteins, they have some domains, and this domain interact with this modified part of uh, the histone tails. Then another protein scent domain containing proteins interact preferentially with unmodified histone proteins or histone tails. So basically, remember just two domains containing proteins. One is bromodomain, one is chromodomain. And this PSD domain, they are generally observed in the plant, but it has been reported recently in the animals too. Fingers specifically recognize modified forms of histone tails. Now, in many instances, remember these things. It is not always if you have the modified acetylations or methylations, this domain containing protein binds always with the all the modified pattern. No, it has been observed that one protein, example HP1, this is a chromodomain containing protein that will bind only to the methylated lysine 9 of H3, only methylated lysine 9 of the H3, but not to any other site of the histone methylations. So some are highly specific to the some sites, and depending on the developmental condition, depending on the disease conditions, the situation is observed in different proteins with this domain containing proteins. There are proteins also reported that, suppose there are proteins that contain PSG finger specific for methylated lysine of pH3 immediately next to a bromodomain capable of recognizing an acetylated lysine. So there are two proteins can bind in the same sites. Now here you can see, suppose this is an histone octamers, and this is here you can see these are the tails, histone H2A tails, H3A tails, H2B, H4, and H3. And this picture is, you can found it in the Watson, J.D. Watson books, the molecular biology of the gene. I have taken this picture from that particular book. So this is here you can see this is the unmodified conditions, and maybe sometimes this unmodified histone core may be methylated or maybe they are acylated depending on the developmental condition or disease conditions. Now when they are acylated, bromodomain containing protein comes and they bind to the particular regions. Here you can see this is the modified acetylated sites. They bind, and according to the need of the cells, they mostly they turn on the gene. Here you can see methylated one. You can observe they are comes chromodomain containing proteins comes bind here, and one beautiful example of bromodomain containing the transcription factors. They generally bind when this bromodomain these factors bind to the when this tail is acetylated. These transcription factors comes bind and they transcribe the gene. So these most of the transcription factors they have the bromodomain. Now here you can see this is the chromodomain, and this chromodomain bind they methylated and they suppress the genes. Now the question, how do the domains that recognize modified histones alter the function of the associated nucleosomes? So one important way is this modified histone recruit enzymes that will further modify the adjacent nucleosomes. Examples, many of the enzymes that acetylate histone tails, this is acyl transferase or heads, include bromodomain that recognize the same histone modification that they create. Okay, that they create. Even this bromodomain or chromodomain, they have the similar functions, and sometimes this particular domain, this modification also can inherit from parents. Now this is the transcription factors. I have already told that this chromodomain containing or methylated containing domains mostly observed in the polychrome group of proteins. You know, they are directly involved in cancers like BMI1 or EGS2. They are directly involved in most of these cancers, and their expression directly related to the methylation of the most of the tumor suppressor genes, and subsequently they promote the cancer. Now, the histone modifications we have just described are dynamic. They are catalyzed by specific some enzymes. Mostly remember this. Very few are inherited. Very few modifications, epigenetic modifications are stable. But most of the epigenetic changes, they are dynamic and they are catalyzed by histone acetyltransferase, histone deacetylase. If from the name you can understand how what is the function of this particular enzymes, HEADs, HDX or HMTs, methyltransferase or demethylases. One enzyme add the acetyl groups, another one remove the acetyl group, one enzyme add the methyl group, or they remove the methyl group. So these are the readers, eraser. These are the function, one function, another function of these enzymes. And currently here you can see this is the summarization. This is also from the latest Watson book. Here you can see what are these type of enzymes. Histone acetyltransferase complex or deacetylase complex we have already known. And most of the drugs we design again the epigenetic changes. We target this particular group of proteins. Here you can see first one is acetylase transferase, deacetylase transferase, methyl transferase, or demethylases. And the detailed function is also there. If you want to study in details, you can refer the Watson book or any other good molecular biology book. You can found this latest published book, means maybe 2023 or 24 after publication date. Now, this identification of acetyl, this is one example. I have not studied it. Please don't take the pictures. Recently, we identified one new in histone 2A, one acetylate amino acids, and we are going to publish it soon. So this is just a technique how we identified it, but through MSMS. Now, is the epigenetic is the epigenome inherited? I have, as I have told you, some epigenetic changes, they are inherited. The genome is passed from parents to their offsprings and from cells when they divide to their next generation. Much of the epigenome is, as I told you, they are dynamics. They are dynamic. They are some part of the life you can observe it, some part of life you don't observe the expression. Much of the epigenome is reset when parents pass their genomes to their offspring. However, under some circumstances, some of the chemical text on the DNA and histone of eggs and sperm may be passed on to the next generation. Now, when cells divide, often much of the epigenome is passed on to the next generation of cells, helping the cells remain specialized. Can the epigenome change? Although the cells in the body contain essentially the same genome, the marked by chemical text on the DNA and histone gets rearranged when cells become specialized. Now, if you want to study in details about this, how this you can I can suggest you one book. This is the Albert book, and how this cell reprogramming happens. Cell reprogramming means they explain it in details. So for this, can the epigenome change? You can found in details about these things in the Albert books. Now, what makes the epigenome change? As I told in one previous figures, there are maybe smoking, diet, infection, disease, etc. Now, another question is, now there are two things. Another thing is imprinting. So whether the imprinting or epigenetics is same, no. Imprinting is a type of or genome imprinting is a form of epigenetics. Okay, not all epigenetic changes are imprinting. So imprinting is just a form of epigenetics. It's a part of epigenome. Now here you can see this when this imprinting or this epigenetic changes, they are or means this wrongly controlled or wrongly regulated, they cause different disease. Here some examples are Beckwith-Wiedemann syndrome, a disorder associated with body overgrowth and increased risk of cancer. So this imprinting, though this imprinting is observed in a normal person also, this is a normal process. Another one is Prader and Willi syndrome, associated with poor muscle tone and constant hunger leading to obesity. Then Angelman syndrome, which leads to intellectual disability as well as motion difficulties. So these are the some of the things which this disease caused due to the abnormal regulation of the imprinting. Now the question is, how this imprinting control the genes, though it is a part of epigenetics. It is a form of epigenetics. I am citing here only two examples. One is IGF2 another gene is H9. H9 is a long non-coding RNA, and IGF2 is what is called insulin growth factor 2. Now, this we know means in most of the mammals or eukaryotes, they have the what is called homozygotes or heterozygotes. They have the duplicate genes, two genes in each allele, or sorry, each gene has a two allele, and one allele from the mother, another one allele is from the father. So here you can see, in some cases, what happens sometimes this imprinting means this expression of gene depending on which parents the gene is inherited, if whether it is from the maternal side or whether it is from the paternal side. So sometimes some genes, they are imprinted or they are silenced or they are activated depending on the which parents the gene has been inherited. So here you can see this is two examples, beautiful examples. IGF2, insulin growth factor 2 gene, and this is long non-coding RNA H19. So here you can see CTCF or CTCT factors which bind here. Okay, what is this? Is the imprinting control regions. Here you can see these imprinting control regions are present in both the genes. This is P means this is the paternal, and M means this is the maternal side. Okay, so there here you can see this is the two genes. One is one allele is from maternal side, another allele is from the paternal side. Again, H19 also from the maternal side and from the paternal side. Now, first we observe the regulation of H19. When it is from the maternal side, what happens? When this H19 is from the maternal side, CTACT factor bind to the ICR regions. Okay, this is the ICR region. This is the imprinting control region or ICR region. ICR imprinting control regions. So what happened? This CTCF bind to the imprinting control regions and they prevent the methylations. Okay, they prevent the methylation. So when the DNA is not methylated, this enhance the expression of this H19 gene in if they are inherited from the maternal side. But what happens when they are inherited from the paternal side, same gene, okay, from the same allele, when it is from the paternal side, they are unable to express why there is no, this gene is methylated and there is no CTCF factor. So this gene is unable to express if they are inherited from the paternal side. Same in the IGF2, they are from the, if they are the maternal sides, they are unable to express. But if they are from the paternal sides, what happens? There are two genes, DMR1 and DMR2. So this DMR1 and DMR2, they are directly involved in the gene expression and suppression. When this ICR region, imprinting control regions, is methylated by DMR1, the gene is suppressed. But when these are methylated with the DMR2, the gene is expressed. So what happens? This if these genes are from the H9 is from the paternal side, they are suppressed. But what happens? This insulin growth factor 2 or IGF2, they are from the paternal side. They these DMR2 bind and they express or enhance the gene expression of the IGF2. If it is the, but in case in maternal side, they are the suppressed. They are unable to express. So these are this how this IGFR2. This there are so many conditions depending on this imprinting. If they are normally regulated, no problem. But if they are abnormally regulated, they cause IGF2 cause different types of disease, from cancer to the neurological to the growth and metabolic disorders. Like this is H19 also cause different type of disease, cardiovascular, aging, oncogenic, and so many things. Now, here you can see now the formation of a cancer cells by epigenetic changes, how they form. If this changes, this is from the EPCOM websites. Here you can see this is the normal cells. Then epigenetic changes, but these epigenetic changes may cause the DNA alterations. So this cancer is initiated, or these changes can cause this is the epigenetic changes, this methylation, suppression, or something like this. They also and finally the cancer initiated also DNA alterations. And finally, these cancer initiation, cancer progression, metastasis, DNA changes, all these causes the cancers. So some of these examples are reported from the journals I have taken from this. How DNA methylation inhibit the tumor suppressor genes, or methylation. Also, this pattern is varies from tumor to tumor to So this is how, this is the expression. This picture is from the Nature reviews. So this, I summarize this from the Lancet Oncology, published in 2002, how this hypermethylation observed in some of these tumor suppressor genes, and hypomethylations observed in the oncogenes. Okay, so this is how this observed. They are summarizing the cell control, invasion, and imprinting, aging. These factors, environmental, hormone, genetics, they are summarized. And I took this picture is from the Lancet Oncology. Now this now the question is food. I have my one of my colleagues, he is working on this particular field, how this food modified this epigenetic and cancers. And here you can see some of the things. This is the cruciferous vegetables and fruits. They contain the quercetin and resveratrol. There's curcumin from turmeric, green tea, EGCG, and ginsetin from the soybean. How they contains or they influence the means cancers, as well as how they are nowadays taking as an adjuvant therapy or no, neoadjuvant therapy or monotherapy against different cancers. The most important thing is that when this epigenome is changes abnormally, they developed either this particular cell is radio resistance or chemo resistance. Chemo resistance means they are drug resistant. Radio resistance means they are resistant to the radiotherapy. And when you take this I will show you one case study recently published in 2022 in my last few slides. So this, when you are taking with the adjuvant therapy or no neoadjuvant or monotherapy, they can chemosensitize or radio sensitize to the cancer or tumor cells. And some of these things here you can see already in published data, they are available. This is in chemotherapy and how they work as an adjuvant therapy. So DNA damaging agents like cyclophosphamide and other drugs, and this radiotherapy, how they work together with the some bioactive phytochemicals. They inhibit the DNA methyltransferase. Some are histone deacetylase inhibitors. Some are microRNA inhibitors. By different routes, they work. These phytochemicals, active phytochemicals, they work, and they chemosensitize or sensitize or prevent the cancers. Now this contribute. This is summarized. I am summarizing it. How do changes in the epigenome contribute to cancer? Changes in the epigenome can switch genes involved in cell growth or immune response. These changes can lead to the uncontrolled growth or hallmark of cancer, or to a failure of the immune system to destroy tumors. So this one example is this is the gastroglioblastoma. Here you can see patient treated with the temozolomide, okay, which kill cancer cells by adding methyl groups to the DNA. When it is methylated, they can preferentially kill the glioblastoma cells, cancer cells. So these are some other examples also there. Now, epigenetics, a personalized approach to cancer therapy. As in our first 2-3 slides, we discussed these epigenetic changes can vary even from the monozygotic twins. Monozygotic twins, they have 100% same genetics, but there are some different characteristics due to the epigenetics. So, from this particular examples, we can refer that epigenetic changes are patient-specific, leading to a personalized epigenetic therapeutic approach. Now, this epigenetic profile of each patient, they are treated with a specific dose of the drug. We can make a profile, and then what happens in each individual, according to the, based on their epigenome of each patient, we can give them the inhibitors, whether it's the DNMT inhibitors or SDEC inhibitors or HET inhibitors. Okay. Now, here you can see this particular region in conventional treatment. Here you can see this is also from one of my students' paper. She is now in USA. So means here you can see in conventional treatment we give the response. So out of these 3 patients, here you can see sometimes all are no response. Some have adverse effects. A few populations they give the response of the drugs. Some population they don't have the response, and some population they have the adverse effects. But when we precisely design the medicines, maybe against the epigenetics or sometimes in genetics also, you can see we may observe the response against all these patients. Okay, those who are not respond to the conventional therapy, they also respond. Now, so this personalized medicine, here you can see this treatment testing. general treatment, then this is called a clinical biological correlation, and this is the omics profile. Based on this omics profile, we can design the medicine in each individuals. Now, one case study, as I told you, I will show you one case study how these epigenetic medicines work. Okay, one case study here you can see this is the a rare presentation of non-Asian. It has been published in 2022. Non-Asian female with this is a study in USA. That's why non-Asian. Non-Asian means she is an African origin. Okay. So with metastatic non-small cell lung cancer harboring EGFR L747P mutations with clinical response to multi-targeted epigenetic and EGFR inhibitor, okay, or inhibition. So this is the first in 2013 August. She reported diagnosed with different conventional medicines. Medications are there. Here you can see this is the osimertinib, then epatinib, and this is erlotinib of the combined epigenetic treatment. They for the combined they are selected a mixture of flavonoid and phenylbutyrate. These are the epigenetic drugs. Okay. So these EP drugs, you can say the EP drugs. Though, so the epigenetic treatment is introduced by intravenous injections. Now, when they combine with this EGFR inhibitor with the epigenetic treatment, from November 13 to 14, patient received several therapies, which included 4 rounds of stereotactic radiotherapy, the paraspinal tumor, radiotherapy along with the right medial lobectomy. Lobectomy means the cut off or removal of an lungs loop for the adenocarcinoma, which was in stage 1. Then chemotherapy cycles with Alimeta and carboplatinum. A laboratory test confirmed the presence of spreading cancerous cells along with the mRNA of the oncogenic protein MYC and the chromatin modifiers HDEC. So immediately they started the epigenetic chemotherapy administered 5 times weekly. The diet plan remained unchanged for that particular patient. In 2015, the researchers lowered the erlotinib dose 25 to 50 gram per day to avoid combination side effects with the epigenetic treatment. In 2015, July 2015, cutoff from epigenetic therapy occurred because the patient could not take further epigenetic therapy, which caused a disease recurrence. Then in July 18, erlotinib resistance was found. Osimertinib has been given for 3 years. Unfortunately, some epigenetic drugs showed some side effects, so an epigenetic switch has been done. Now here you can see in July 2020, a localized radiation was done on the pulmonary lesions. A disease progression was again found during that time. Osimertinib at the dose of 80 milligram per day was going on. After the laboratory test, the epigenetic protocol was placed for the patient, and on the treatment with epigenetic, the patient decided to reduce the epigenetic therapy to once per month, which causes disease increasement. Now, June 21, after molecular analysis, advancement of gene mutations have been found in TP53, AKT, and retinoblastoma. Therefore, they were restarted the epigenetic therapy combined with decomitinib of 45 milligram daily on a 5-day on-off schedule. Epigenetic therapy was continued 4 times per week for 6 weeks. The treatment protocol was completed on April 23, 2021, and laboratory investigation were conducted on 15 June 2021. Result, the summarization of the result is, in this study, epigenetic therapy combined with the low-dose treatment targeting EGFR has demonstrated a very good progression-free survival with more than 8 years of life expectancy and no severe or chronic side effects. So here you can see a 56-year-old non-Asian woman with oligometastatic NSCLC. Before study, various types of treatments were there, as we discussed. Based on the biomarker detection, patient was treated by epigenetic therapy. Now, then treated the patient with EGFR inhibitor plus epigenetic therapy. Treatment only with in a few months. Treatment because of the side effects. The treatment only with the EGFR inhibitors. Again, treated patient with epigenetic therapy and plus EGFR inhibitors, and this one is here you can see 8 years survival without suffering from any serious side effects. So we don't know whether she is alive now or not. So this was published in 2022, and now 2025. If she is alive, maybe it means more than 8 years. Okay, okay, 8 4 means almost 11 or 12 years. Now the conclusion is, in recent decades, personalized epigenetics has been gaining more and more attention from the researchers for its fine application and impact on disease. In numerous clinical trials, epigenetic therapy has demonstrated a remarkable therapeutic result when treating patients with different forms of cancer. Accumulative evidence showing its importance of epigenetic therapy in the field of cancer. However, several combination therapy trials are needed for this personalized epigenetic treatment to make a firm base in cancer treatment. The mentioned case study showed that combination therapy with epigenetic treatment might be one of the choice for treating cancer patients. In the future, a greater number of clinical trials will be needed. It is important to carry on this type of combination therapy for better advancement in personalized epigenetic therapy. And these are the some of the citations. Thank you. Thank you very much for your attention.
Thank you, sir. Thank you very much for this informative session.
If you have any questions, you can ask.
So there are a few questions in the chat box. I can read it for you.
Where is chat box?
Hafsa Arif has asked, sir, what strategies can overcome poor bioavailability and pharmacokinetic limitations of epigenetic phytochemicals in clinical approaches?
Yeah, there are so many things are there. I mean, say this poor bioavailability not only problem with the phytochemicals. This poor bioavailability also we observed in some other synthetic drugs too. But scientists are working. This is one thing is that this nano medicines or nano delivery or formulation of nanoparticles of these particular drugs. So this is one of the solutions. Yeah.
So there is another question by Nilofer Banu. She asks, how do epigenetic modifications such as DNA methylation, histone modifications, and non-coding RNAs contribute to the development of cancer drug resistance? And how can these changes be therapeutically targeted?
So means there are so many genes are there. Those genes are directly involved with the chemoresistance. Suppose polycomb group of protein. This is one polycomb group of protein. This is BMI1. So what happens when this most of these oncogenes, what happens? They are acetylated. They are overexpressed. They confer the chemoresistance. So this is the epigenetic changes. Though the DNA sequence, all these oncogenes are not mutated always. So they confer when they overexpress, they confer the chemoresistance. Okay. Similarly, in tumor resistance or tumor suppressing genes, they are when methylated, they are suppressed. So they are unable to cope up with the tumor load. So by that way, these epigenetic changes can either promote or they can suppress the cancer. Is it clear, Hafsa? No, this is Nilufar Banu, I think so. Nilufar Banu, is it clear?
Yes, sir, she has responded with a thank you, sir. Their mic has been muted, so they are replying in the chat box.
Oh, okay.
There is another question by Khalida Siraj. She asks, well, will antioxidants present in epigenetic photochemicals not lower the effect of chemotherapy?
Antioxidant present in epigenetic phytochemicals. There is a beautiful articles I have published in 2023, whether these phytochemicals are four or friend in oxidant antioxidant cycles. You can read it. This is publicly available. Okay, these are free accessible. Where we can example so many examples I have. If Ms. Khalida, if you just give your mail ID to me, I will forward you this article. There are so many examples are there. Okay. So we have put whether they are sometimes they are for these phytochemicals, sometimes they are friend. Not always these phytochemicals are anti-cancer activities. Sometimes some phytochemicals they also promote the cancer activities. And we have listed a group of phytochemicals in that particular paper. It was published in 2023. So it is not very old. It is a very new article. I will forward to you. You can find the details, how they work, what is the mechanisms, how they promote the cancer, and sometimes how they inhibit the cancers.
Okay, thank you very much, sir, for a wonderful talk. Now it is time for a short break, and let's gather us all after lunch at 2 p.m. And Sir Khalida has shared her email address in the chat box. You can find it.
Yeah, you can. I have also share my. If someone has some questions, they can ask me.
Okay. Okay. Thank you very much.
Thank you. Thank you. Thank you very much.
Thank you, Dr. Hifzur. I would request you to please provide the slides for the participants to be accessible. So I'll upload it on the GAN website in the Google Drive that we have.
Okay, sure.
Thank you. Thank you so much. Thanks to all the participants who are here, and we'll see you at 2 p.m. with Dr. Asim.
Hopefully, I am on time now. 1 hour.
Perfect.
Yeah, thank you. Thank you very much for giving this opportunity.
Good afternoon, everyone. Swati, I'm not able to share my screen for some reason.
Yes, so one moment, please.
Thank you.
Can you please check now?
Yeah, I can do that now. Thank you.
Okay. Good afternoon, sir.
Thank you. Very good afternoon to you as well.
So shall we begin this session or should we wait for a few more minutes?
Totally up to you.
Okay, people are still joining, so we can wait for 1 or 2 minutes.
Sure. In the meantime, I can check whether my screen is visible. So can you please check if you can see full screen and you can see my laser pointer in red?
Yes, sir.
Perfect. Thank you very much.
All right, so welcome back, everyone. I hope you enjoyed the break. It is once again my honor to welcome Dr. Mohammad Asim, who will be presenting today on the topic metabolic vulnerabilities in cancer. Thank you very much, sir, for joining us. Over to you.
Thank you, Swati. Um, Likewise, pleasure to meet all of you today again. Um the structure for today is that we're going to talk about the metabolism, and especially with regards to the cancer cells. Sorry, my voice is a bit croaky today. Flu, not wanting to go, um, but hopefully I'm audible clearly. Uh, and in addition, what we're going to talk about later is some work, some research, latest research from my laboratory, uh, as well as from other laboratories on solving the problem of prostate cancer. So, without any delay, I'm going to talk to you about the uh, metabolism that is experienced by cancer cells and how you would start to appreciate differs from the normal cells. So have a look on your screen. What you can see here is basically central metabolic pathway, which is glycolysis. And then, of course, this is what we are taught in BSc. Um, Mostly those of you doing biochemistry know this pretty well. And then you have the Krebs cycle. And what happens here, the reduction of these NAD and FADH is then oxidized again through the oxidative phosphorylation cycle, which generates ATP. That's how the metabolism, as we know, works in normal cells. It has been noted that cancer cells are actually quite acidic. Where they are formed, the cancer is formed, they make sure that their surroundings become acidic. Now, why would that happen? Uh, why they are making and secreting acid in the nearby environment? One of the things which we now know is that it can actually stop the immune attack. As you know, cancer cells, because of the mutations, they express some proteins which are missing on normal cells. So these proteins are somehow changed in their shape thanks to the mutations. And that should make them visible to the immune system, right? But cancer cells can actually circumvent this. They make sure that even if they express these new proteins, what we call new antigens, uh, these are essentially coded by the genes which we already have. They're not new genes, but these genes, because of the mutations, have subtle changes within the protein structure that they give rise to due to mutations. And as a result, they should at least in theory become perfect target for immune attack. It does happen in some cancers, which are we were discussing day before yesterday, who have the MSI or microsatellite instability, something a pathologist or a genetic technologist in many countries can have a look. They can do karyotyping, look at uh, the chromosomes, but also electron microscope studies, and most importantly, sequencing will tell you whether there are microsatellite instability in some chromosomes. So this is very important. Cancer cells want to achieve a lot of things. They want to spread, they want to invade, they want to melt the matrix where they are present. And at the same time, now you realize that actually they are carrying out an altered metabolism so that they can enhance the production of acid, specifically lactate. And that allows them to evade the immune attack. So these cells are not really something which immune system will be able to see and attack them. And there are multiple advantages, which of course, these are textbook advantages. Uh, what I'll try to do is give you a brief overview of how it works and what are the key advantages to cancer cells, essentially tumors. I should also point out that when you're growing cells in a cell culture dish, you may not actually see many of these phenomena happening. The reason is very clear. Cells do not experience the same type of environment when you grow cells in cell culture dishes. You are growing just cancer cells. They don't have an immune microenvironment, but also they are not growing in 3D. They don't have any problem in getting oxygen supply. Why am I saying oxygen supply? Because as you would see that many of these pathways which allow cancer cells to secrete acid in their environment, they actually are carried out through glycolysis. And what happens in glycolysis is shown on your screen that cancer cells prefer. So normally what is happening here is that this pyruvate which is produced usually, and I'm talking about normal cells, it is converted into acetyl-CoA, goes into Krebs cycle. If a cell experiences that there is no oxygen or there is less oxygen, they actually decrease the flow of energy generation through this pathway, but they start relying on these pathways. This happen in normal cells, which cells, muscle cells, intensive aerobic exercises, we start getting cramps. And this happens because, as you would know, generation and accumulation of lactate. But what is happening here through generation of lactate, ethanol in other cell types, other organisms under oxygen depletion, is that you generate these acidic molecules in a cell. And as a result of this, what is happening are two things. One of them is that you don't need to generate ATP through Krebs cycle and through oxidative phosphorylation, which is, of course, a longer process. Can you think of which pathway would generate more ATP? Do we generate more ATP through glycolysis or through Krebs cycle and then oxidative phosphorylation? The answer would be, of course, through Krebs cycle and then oxidative phosphorylation. That generates much more ATP than glycolysis pathway alone. And it seems that cancer cells prefer this pathway, glycolysis. So looks like they're a bit silly in that they don't really use a pathway which is much more efficient in generating energy from glucose molecules, but they rather rely on glycolysis. One of the advantages I just told you of you relying on glycolysis is that they can make their environment acidic. But there is something more happening here. I showed you 2 days ago a picture with birds during migration. Uh, they were prepared to do that migration. Exactly something similar is starting to happen here as well. These cells, when they're small tumors, they have sufficient oxygen supplies. As I told you the other day, tumors become bigger and they can actually try to invade, but also they can try to make new blood vessels to gain sufficient blood that can bring nutrition, right? So what these cells are trying to do is they're preparing themselves for an imminent lack of oxygen, because when solid tumors grow, many of them will not have sufficient oxygen, especially if you see, if you take a cancer biopsy and look at the internal cores, they will have very high hypoxia. They will not have sufficient levels of oxygen within them. And one of the reasons some cancers prefer to sit in metastasize to brain is within our body there is a different concentration of oxygen. Our lungs usually will have very high levels of oxygen. They are in direct contact of it, right? But as opposed to that, our brain has lower levels of oxygen. I'm not saying it does not, but if you look at the gradient, there is, of course, more oxygen in lungs than brain. Now, these cells are getting ready for a move. They don't, at this diagram, what you're seeing, they don't really need oxygen. They're trying to work out a metabolic pathway that can allow them to sustain without the need of using oxygen. Brain has less oxygen as well compared to lungs. And there is a whole gradient. You can have a look at that table. So what is happening is that these cells would also prefer, they would be perfectly fine sitting in brain and growing. And as we discussed yesterday, brain metastasis actually is super bad when it comes to the patient prognosis. So these cells look like they're very good in many aspects. They can carry out lots of acrobatic exercises. I was telling you, they move from primary tissue, they can go to other organs. But they're not very good at making use of glucose, it seems. Why? One of the reasons, again, is that they can make the environment acidic. And they also learn to live without oxygen. Uh, that is because they know eventually there will be a threat coming that will allow them to experience the deficiency of oxygen. So they're preparing for future like humans do, preparing for future. In this case, they're preparing for their future that they're going to experience deficiency of oxygen. So let's start not using oxygen and rather rely on this particular pathway, glycolysis, without the need of using oxygen. So if I show you an example, please feel free, by the way, to ask any questions if you have via the chat box, but I'll be more than happy to take questions afterwards as well. Swati, for some reason it is saying continuous chat is off. So could you kindly switch on the provision that the attendees can ask me questions if they have any. So that's clear, hopefully. So have a look. This is something which was noted by a scientist called Otto Warburg, that cancer cells actually use a pathway which does not really rely glycolysis, which does not really rely on the availability of oxygen. So have a look with regards to the energy generation, what is happening here. These are differentiated cells. You can think this of being part of an epithelial cell layer. And what is happening here is Swati. Can you hear me?
Yes, sir.
Okay, perfect. Yeah, I think the chat is off for some reason. So students or attendees may not be able to ask questions. They cannot type. Let me send a message
Chat
saying
is
test.
still on for the participants.
Okay, perfect. Thank you, Padmavati. Okay, so back to this topic again. So what is happening here is that normal cells usually they have good access to blood supply and therefore oxygen. And that allows these cells to carry out generation of energy, i.e. ATP, through the mitochondria, it Krebs cycle, and then oxidative phosphorylation, which generates a lot of energy per glucose molecule, as you can see on your screen. What is happening here that these cells, when they don't experience, I'm talking about muscular cells undergoing extensive aerobic exercise, they will not have sufficient oxygen available to them. And as a result, the system, the metabolism will be altered, leading to the production of lactate, again, which is acidic. In normal individuals, it will cause muscle cramps, but in cancer, these cells are very clever. They're making sure that nothing comes and nothing that can destroy them, especially the immune cells, that they will be deterred. They won't come anywhere near those cancer masses, seeing that they have their microenvironment. The environment where they're present is actually quite acidic. So as you can see here, this pathway is not very energy efficient. You generate far less molecule of ATP per glucose molecule. So if you see proliferative cells or tumors, there is something common here as compared to these differentiated tissues. So both proliferative tissues which are dividing very, very fast, perhaps at par with tumors, but then they are still normal tissues. Can you give me some examples of some tissues which grow very, very fast? These are the cells which are badly hit by chemotherapy. So the cells of our hair follicle, sorry, I don't have longer hair this time. So the cells in our hair follicle, they are one of the fastest growing cells, but also cells in our gut, they divide very, very fast. And therefore, you may notice cancer patients who undergo chemotherapy will lose as a result of therapy. Chemotherapy will kill any cell which is dividing very, very fast. For example, tumor that is the intended effect, but they will also have side effects because they're also going to kill other cell types which are growing very, very fast. So fast growing cells, especially cancer cells, what happens here is a very little of it, actually pyruvate, goes into mitochondria, and very little of it actually forms is gives rise to through oxidative phosphorylation ATP, but the majority of it is still making a lot of lactate. So it looks like cancer cells are not, after all, very, very energy efficient. They're rather very poor in this process. So if I show you some energy ratings of cancer cells, have a look here. They will perhaps be here. Their energy rating is really, really poor of cancer cells, thanks to their reliance on aerobic glycolysis. So they need, of course, they're growing much more faster than many. Sorry, they're growing much faster than many cell types, normal cell types, and at the same time, they're not making good use of glucose. So what happens as a result of this process? They can generate energy much more rapidly. Why? Because they're relying on something, glycolysis, which they can carry out in the cytosol. There is no need to involve mitochondria or the oxidative phosphorylation. So the generation of energy is much more faster. But also there are intermediates of glycolytic pathways which can actually take part in many synthesis reactions, anabolic reactions. Why? Because cancer cells are dividing much more faster than a normal cells. They will need a new fabric, they will need raw materials, and this raw material comes from many glycolytic intermediates. What I also mentioned is that by making and secreting lactate, it allows them to survive in a low oxygen environment, but also they can suppress the immune system attack through this. And lastly, but very importantly, what also happens when you have oxidative phosphorylation through the electron transport chain in mitochondria, that is the primary source of the production of reactive oxygen species. That is what is used by many therapies, including radiotherapy, to kill cancer cells by destroying the DNA. So if you can suppress the oxidative phosphorylation, that is what happens in aerobic glycolysis or Warburg effect. There is a less concentration of reactive oxygen species at this time, meaning the cells will survive this. They will not have very high levels of reactive oxygen species. I was telling you 2 days ago, there is something else which can also dampen down, which can also reduce the level of reactive oxygen species, which will be pro-cancer, which will promote cancer. Um, Antioxidants. So taking a lot of antioxidants in its super high, ultra high form uh, artificially may not be a very good idea for general population uh, themselves. So we should be very careful with that as well. I hope this is clear so far. So these cells are needing, they are growing much faster, but their system to generate energy is not very efficient. They're generating energy through glycolysis, but converting most of it into lactate, which is secreted. The rest of it is basically the source of uh, energy. You can see per glucose molecule compared to normal cells, they're making far, far lesser energy. But who needs more energy? Differentiated cell types or cancer cells? Well, cancer cells need more energy. So how do you compensate this? You compensate this by enhancing the level of glucose that you drink. So we know cancer cells are super hungry for glucose for this reason. They need to divide really, really fast. They're not very energy efficient. So what they start to do is to steal glucose from their environment. So they become highly, highly competent in taking up glucose from which was intended for other cell types. So have a look at this PET scan. What you can see here under normal circumstances, we have tissues which are very intense. They have very high energy demand. And if you see them, these are the PET scan, FDG PET. So the glucose has been labeled, and that will show up on a PET scanner labeled glucose. And you can see here, if you do a PET scan of a normal individual, you can see here what cell types, what tissue types are actually the ones consuming the most glucose. So our brain, our heart, no doubt, as well as the transition, because of the transitional epithelium structure, which is very flexible, the bladder seems to make use of a lot of energy. If I show you this slide, sadly, of a patient who has ARMS, the type of cancer of the alveoli, you can see here, in addition to the heart, brain, bladder, the cancer cells are actually lightened up by FDG PET. All these cells, what you can see here, they are consuming a lot of glucose. So this is actually what forms the principle of detection of cancer foci, not the primary tumor, but you can see even the little metastatic spread of tumor, where this is gone. Has it gone into liver? Has it gone into the pelvis area? Has it gone into the vertebra? And you can see, sadly, in this patient, it looks like cancer has actually spread a lot, including the bone system as well. So by exploiting the ability of cancer cells to take up a lot of glucose, you can actually help in the staging of cancer and localizing cancer as well, where is it located. And this information can then be used by radiotherapists to actually destroy through laser guided, with laser precision, where this cancer cells are and whether they can get rid of them. Uh, does it make sense? Any questions so far? Well, if there aren't any, then let's carry on. Okay, so what we discussed so far is the ability of these cells, cancer cells, to make use of a lot of glucose. Why? Because they want to do a lot of things. They want to make their environment really poisonous, i.e., acidic. They want a lot of glucose because their energy demand is far, far higher. Uh, and That's why they start to compete with other uh, cells and tissues, glucose, which was intended for them. And therefore, one of the things that happens to cancer patients is muscle loss, weight loss. Cancer patients will become really, really skinny. Many of them, in fact, one of the things that doctors, when they suspect in many countries, including India, substantial weight loss over a short period of time. Sometimes there may be suspicion of cancer taking hold as well, because of the fact that these cells are actually consuming a lot of glucose and therefore depriving normal cells of their diet, i.e., glucose. And in doing so, they actually cause sarcopenia, so loss of muscle fibers, muscles themselves, causing patients to lose a lot of weight. So they're using glucose as well, but as you would see, what can also happen in these cells is they become highly, highly metabolic, plastic. And what I mean by that is that normally what will happen, you can consider this, these are primary tumor cells, and they're sitting here on top of the basement membrane, and then they take a journey where they invade the connective tissue. They can intravasate to get hold of the blood vessels, and they go into the blood circulation system. We've discussed this before. I'm just revisiting it to give you a recap for the sake of this lecture. And what happens here, these cells, when they reach their destination organ, these cells can extravasate and start forming colonies in secondary organs, for example, lungs and liver. Bone, of course, is included as well. And what has been noticed that these cells, throughout their journey, they can actually change the requirement of nutrition. For example, these cells may rely primarily, while they are within this matrix, on glucose, but it has been noted that cells which are in circulation may have different need. They may change the requirement of the carbon source for as a nutrition. And you can see here they become more flexible in terms of the metabolic pathways that they can switch on, and therefore what nutrients can they use. So you can see, for example, nutrient 1, 2, 3, they can actually make use of them while they are in their journey. When they come back to their target organs, they may again switch back to using their original carbon source, which is glucose. As we discussed, all these cells, what you see, these black dots, is nothing but cells which are drinking uh, a lot of glucose, making the cancer foci visible on a PET scan. So if I show you the relative plasma levels of some metabolites, some carbon sources, have a look. Healthy individuals usually will have very low levels of pyruvate, but it has been noted as individuals develop cancer, their circulating pyruvate level increases. And in metastatic disease, in breast cancers and others, they have enormously high level of pyruvate, circulating levels of pyruvate. And same is true for lactate as well, especially in colorectal cancer. You can see an inverse correlation in glutamine and glutamate in esophageal squamous cell carcinoma. And what you're seeing here is there is a primary requirement of glutamine in primary cancer, but as cancer progresses, the requirement of using glutamine as a nutrition source may be dampened. And the opposite is true for glutamate. Normal or primary tumor cells will not have very much in the patients will not have in the plasma very high level of glutamate, but later stages, especially metastasis, will have very high levels of glutamate. And likewise, similar is true for colorectal cancer and breast cancer and many other cancer types with regards to lipids. We know that one of these nutritional sources that these cells can use is actually none other than lipids. They can make use of lipids as a carbon source to generate energy from them. So these patients will also have very high level of lipids in the body. Now, what is the whole point of all of this? They're making use of these compounds. We are able to measure a handful of these metabolites in the plasma. There are people who are now thinking to detect cancer early, because if we know this full well, if you can detect cancer early, you can have better ways to treat it, and sometimes the treatment can be curative, meaning that cancer would not be likely coming back again, and they will die of some other cause than cancer. For that, we need to detect cancer early, and for that, what we need to do is collect samples from normal individuals uh, prospectively. So what has been happening, at least I know a few trials, including the city where I live in, Cambridge, is that there are many GP services who allow you to give you a sample of urine as well as blood. They ask you for 1 ml extra blood, and that will be preserved. Same happens with urine. They will be able to freeze it and keep it there. What they want to do then, because they know that the population of the city is quite stable, there aren't very many people coming, and if people come, they usually live there for a decade or so. What they want to see is, okay, we are collecting the blood today, and we will follow up these uh, individuals to see how many of them develop particular type of cancer. So I'm not talking about 10 people on trial, I'm talking about thousands of people on these clinical surveillance studies to see which of them develop cancer. Then you can go back and have a look of the samples which was collected 20 years ago and measure the metabolite levels and see whether there are any patterns emerging with regards to the levels of these are 4 or 5 metabolites I'm showing, but actually through the chemical analysis you can measure hundreds of metabolites in plasma samples and urine samples. This is exactly the intention uh, because of which they're collecting a lot of samples these days to be able to go back retrospectively and test for whether they developed any cancer or not, and whether there were any anomalies, there were any metabolites, even 10 years, 20 years before the cancer became manifest on a scanner, for example, or manifested itself uh, clinically, that they can actually make any interventions going forward for future generations. So what we can do in scenarios like, of course, India is we have a big population that is positive. So if we can collect those samples from patients, not patients, normal individuals, and keep them. The challenge would be, of course, to keep them in -80 C, which requires a lot of energy itself. But if we can keep them certainly and start such clinical studies, we have a big positive, because the populations in cities is really very large, and even if you can get a fraction of people to take part in such trials, Actually, that would be very good in terms of detecting cancer early and perhaps make predictions for if somebody comes and we found out metabolite X was associated with colorectal cancer in younger patients, then perhaps those patients can have scans, endoscopies. Uh, they may be put on surveillance earlier if there is a strong association found with metabolite X and the diagnosis of colorectal cancer. And the cancer may be removed at a much earlier stage when it is a primary disease, really tiny tumor compared to metastatic tumor, of course. So that will certainly save precious lives, but also it will be, you know, good for you as research scientists to devise, uh, you know, protocols, clinical protocols like this that will allow you to do that. This is not something which can be easily done in Western countries, thanks to the decline in the population, aging population, but also the cost to the health economies. It's very expensive in these countries to even buy things like a simple proton pump inhibitor. I think 7 tablets you get is about 500 rupees equivalent. So there are good things in India that can be done, but of course, for that, basic scientists would need support from clinicians while they're taking blood, whether they're comfortable, of course, giving patients the information sheet, whether they would want to partake into clinical trials and give 1 ml of extra blood and a urine sample. So, of course, these are some things which we can do. Trust me, we have very good systems. We have good surgery and we have good intervention uh, for cancer. The problem with most cancers, which kill human beings, is that cancer is usually detected very late. For example, pancreatic cancer, it doesn't really say anything. There aren't any symptoms until it has a spread. And that's why for pancreatic cancer, the incident and the mortality is almost same. Within 5 years, it will kill 95% of the patients. So let me scientists have been able to devise molecules based on the ability of cancer cells or their hunger for glucose, small alterations in glucose molecule itself. So what you're seeing, this is a glucose molecule. What has been changed here by chemists is this hydroxyl is missing here. So now this is not glucose, but it's this 2-deoxy glucose, like deoxynucleic acid. This is 2-deoxyglucose. So what happens with this molecule? If you use this as a carbon source, of course, this is taken in by the cell through the glucose transporters and is actually recognized by the first enzyme, the hexokinase. And hexokinase can phosphorylate this and traps it as 2-deoxyglucose 6-phosphate. Well, hexokinase traps glucose in the same way. It phosphorylates glucose, so it cannot leave the cell. Likewise, what is happening here, 2-deoxyglucose can actually be formed by this reaction, 2-deoxyglucose 6-phosphate. Hexokinase does not really discriminate between glucose and 2-deoxyglucose, but once 2-deoxyglucose is formed, it can actually shut down this whole glycolytic pathway. So that is something you can think of using as a treatment for cancer, which are highly dependent, and in fact, majority of solid tumors, I said, are reliant on their ability to suck up a lot of glucose from the environment because of the Warburg effect. So experiments have been done with this 2-deoxyglucose where this has been given to animals, and I give you an example here. These are animals, mouse, in which the tumors are grown, and then they are given 2-deoxyglucose as a treatment. And look what happens here in these mice which have tumors. They can grow. You can see on how this is y-axis, the tumor volume, and how many days does it take for the tumor volume to for the tumor to achieve this volume. I'll start taking questions in a minute. I have about 6 minutes, then after questions. Khalida, please hold on. Um, I've seen your question. So what is happening here? 2-deoxyglucose, when it is given to these animals, you can see here the tumor is completely flattened out. It does not grow as good as the tumors in the other arm, where there is no 2-deoxyglucose given. The if you take out those tumors after the study is complete, you can see here tumors in animals which did not receive the 2-deoxyglucose, the metabolic poison I showed you on the previous slide. These tumors actually grow much bigger within 2 weeks. But have a look here at tumors which were given 2-deoxyglucose. These tumors are really, really tiny. Uh, mm, because of the fact that they are not able to grow as fast. They need glucose, so they will suck up 2-deoxyglucose thinking it is glucose, but in fact it is not. And as a result, you can see this can be achieved at least preclinically. The experiments that we do in animals, we call them preclinical experiments because they have not utilized any human being for the treatment. And for starting any clinical trials, at least up until now, for cancer therapy, you will need strong evidence that your treatment works in animals. At least 2 different species. One of them is mostly mouse, laboratory mouse. The problem with such therapies might come is whether these metabolic poisons are safe or not. There have been issues identified when these were given to animals. Sadly, uh, many times what people do when they're publishing papers, they only look at what I'm showing you on your screen. The growth of the tumor is affected or not. They don't see the welfare of mice. They don't even do, many of them, blood profiling to see whether there are any changes in mice. All they look at is the basic phenotypic changes, the behavioral changes in animals who have been put on these new drugs. But that's not sufficient evidence to take them into clinic. The drug regulators, clinical trials regulators, would want to see the data, extensive data of the side effects on animals before giving a green light to carry out such studies in human. And of course, the metabolism in human and a mouse are different, and because of that, treatments may not work in the same way. Does that make sense? So I will stop after this next slide I'm going to show you. This is quite interesting. Again, some changes happening into the uh, metabolic pathways, giving rise to new things. For example, if you have these aerobic glycolysis, you see that these cells become super hungry for glucose, right? You saw that in the previous example. In this case, what is happening, because of some alterations, some mutations, to be precise, cancer cells start making some metabolites which are missing in normal human beings. One such metabolite is called 2-hydroxyglucose. Now, what happens in Krebs cycle, if I show you here, especially this step, is that you have this isocitrate, which is converted into alpha ketoglutarate by the enzyme called isocitrate dehydrogenase, or IDH. The mutations in IDH are one of the commonest in multiple cancers, especially glioblastoma, which is a specific type of brain cancer. Several years ago, I did some transcript analysis in prostate, and we found one of the top mutation and overexpression in the data set was none other than IDH. So IDH was mutated in cancer. Now, this looks quite simple. because of the mutation, the although the substrate is still isocitrate, the product which is formed through these reactions is not alpha ketoglutarate, but it is actually a slightly changed chemical entity. It is 2-hydroxy glutarate. Now, 2-hydroxyglutarate. So again, what is happening here? What the impact of this will be that the TCA cycle will be shut down or will be reduced. Why this is happening? Because cancer cells are trying to get ready for the imminent, for the near future loss of oxygen. They know that the electricity will be gone and they will be going in the dark. So get your torches ready. Likewise, what they're trying to do here, they know there is no oxygen going to be available. They cannot really rely on oxidative phosphorylation. They cannot rely on TCA cycle. So switch on glycolysis first thing first. They've done that. Okay, any residual acetyl CoA coming into the TCA cycle, block it. How do you block it? Block it by making mutations in the isocitrate dehydrogenase pathway so that you don't continue with this TCA cycle. In fact, make 2-hydroxyglutarate, which I'll tell you in a minute why this is important. What happens to 2-hydroxyglutarate in terms of the fitness of cells under low oxygen conditions. It can block many enzymes. As you can see, many of them are epigenetic enzymes. I'm not going to go into the details of epigenetics, but 2-hydroxyglutarate is called oncometabolite. It is one of those metabolites which is not found in normal cells. And this is something which can be detected through the blood. So you really don't need a lot of tools in order to detect this. You don't need to do biopsies. You don't need to put patients under anesthesia or have multiple doctors operating on a patient. You can, in order to diagnose, you can simply detect it through the plasma. And this is secreted by cells which have IDH mutations. The challenge for those of you who are a student is to identify more oncometabolites. Um, that's not what we're very good at. We just don't know what sort of metabolites are formed. Oncometabolites are formed by cancer cells. There are only few known, but nature is not silly. They're making them for some specific use. In this condition, so far what we have discussed is they're preparing for an imminent loss of oxygen, because as they grow, they're going to experience the loss of oxygen, and they're preparing it. They cannot rely on TCA cycle, so they're starting to enhance the level of aerobic glycolysis, what we call Warburg effect. They're shutting down this metabolic pathway, and then they're making this 2-hydroxyglutarate, which will help them again with the survival under conditions of oxygen deprivation. Who wants a break? I know I've gone through 45, 47 minutes. Dr. Aziz, shall we take a break? I know you're running late in terms of you're 5.5 hours ahead. If nobody wants a break, then I can continue to talk until I finish this. Totally up to you. Please let me know. Okay. In that case, I'll stop and see some questions which have been asked in the meantime. Those of you who want to take a break, you can take a break and let me have a look. So Khalida is asking, I've seen an old age person suffering from bladder cancer having all gray hair, undergoes chemotherapy as well as radiation therapy. After the treatment, he lost all his hair, but after some time when new hair grew, they were all black. What is the reason behind that? Really, this is news to me. I know that the people will grow back new hair. I don't know the reason why they grew totally black hair, but I will certainly check on this, whether this is something that happens as a result of chemotherapy or radiation therapy. They do, the hair do stop growing, not as a result of radiation therapy. If it was given through EBRT, external beam radiation, that will not cause such side effects. These are the side effects which should come from being on chemotherapy. If they had bladder cancer, I'm sure they must have undergone treatment with BCG, uh, possibly. But if they were on chemotherapy, of course, they will lose hair for when they're on the treatment. They eventually grow back. I was not really, this is news to me that the hair was black. Any other questions, comments? You can switch on your mic. Feel free to ask me questions if you have any. Sunday when I was asking, I came by a recent article where it was estimated how intermittent fasting is reducing cancer risk by starving cancer cells. Is it related to metabolic starvation or any other correlation? Very good question, Sunday fun. So there are, of course, benefits known of metabolic fasting. Some of them, sorry, not metabolic fasting, intermittent fasting. Some of them, in fact, come from the ability of cells to undergo autophagy. This is very well known. A Japanese scientist received a Nobel Prize a few years ago on this discovery of autophagy being promoted under conditions of nutrient starvation. This is something we already know. Sandeepan, we have to realize humans, as we know, they were not really had uh, food like breakfast, lunch, dinner. We were wandering in jungles in prehistoric times. The food was not kept in freezers, was not sold in supermarkets, and it was not frozen. Of course, they had cancer, as I showed you, but perhaps the cancer was not as common as it has become now. And one of the reasons was that those people we know, they starved. They didn't have so good access to food as we do now. Yeah, with regards to intermittent fasting, we know there is certainly benefit of intermittent fasting. And we also know that mice, which are calorie restricted, from the same genetic makeup, if you have mice which are calorie restricted, sometimes live twice as much as mice which have been fed with high fat diet, for example. So we know calorie restriction does help in longevity, definitely. Any other questions? Or we will start in about 8 minutes again. So feel free to ask me any questions if you have. Uh, well, plasticity, simply, these are overlapping terms. But basically, what happens is metabolically plastics means that they are able to not only require, they are not absolutely dependent on one single nutrition for their survival, but they can use multiple sources. For examples, when we were living in jungles, what we ate as human beings, mostly roots, right? Those roots are really, really very super solid, very hard to chew. And you see many skeletons that we discover through excavations. Many of them have had problems with their tooth. 20-year-old male with many molars, premolars missing, trying to chew food. Some of them actually had a, we know that died of uh, infections, excruciating pain, going into their jawbone as well. Dr. Gupta can probably shed more light into this. But basically, we had a different food back then. Now our food is totally different. So you can see we are, as human beings, as organisms, we are quite metabolically plastic. What we can eat in terms of nutritionally plastic, should I say? So cells also, in order to become fitter, they can actually also use different varieties of nutrition as carbon source. And sometimes, because of these changes that they are going through, those changes make them fitter to survive, because you might find that some carbon sources are much more abundant in blood compared to solid tissue. So they learn to make use of this. And thanks to our pathways, metabolic pathways, which actually allow them to be used as well, because these nutritions, these carbon sources can enter through glycolysis pathway, also through TCA cycle as well. I hope that answers your question, Mustafa. Great. Dr. Aziz, just taking 4 more minutes for anyone to ask, and then we'll reconvene. Sandeepan is asking, there is a dietary concept of sugar coating, sugar cutting, and promoting natural sweetener like honey and jaggery, replacement for cancer patients and patients undergoing remission. But I see glucose is the main culprit of tumor cell promotion. Is it still valid statement? Um, Sandeepan, it's complex. It's an excellent question you've asked. And with regards to sugar cutting, certainly this might help. There are trials where patients have been asked not to have any sugar at all, especially the sugar with no nutritional value, what we call, you know, uh, only calories, pure calories, nothing else. With regards to jaggery and honey, of course, these are more nutritionally good. My suspicion would be, Sandeep, on the way our own immune system kills cancer cells is by many times generating these reactive oxygen species that need to be there for even cells during the immune attack. Uh, with super low levels, ultra low levels of glucose, how this is going to function. So these cells may actually be protected against immune attack. Maybe there is already some literature on this. I don't know. But with regards to, so many of these dietary interventions actually work more in settings of chemo prevention. Chemo prevention is something like chemotherapy, but chemotherapy you start after somebody has cancer. Chemo prevention, I would say, refers to something where there is still time for cancer to progress. You know that somebody in the family, the mother, father, developed cancer, perhaps died of it, sadly. So you can actually do this intervention 20 years before that, okay, this is very common in my family, prostate cancer, for example. Okay, I know epidemiological data suggests that patients who have very high dietary consumption of dairy products, they are more likely to develop prostate cancer. So those individuals may think of cutting dairy consumption from their diet as such. With regards to jaggery and honey, I do consume. Well, this morning I had tea with honey in it. It may not be suitable for many people to have a you know very high fructose diet. I would say I would not take into my body anything where there is no use of that substance. For example, these artificial sweeteners and so on. I'm not sure how they are metabolized by the cell. For glucose, I know there is a proper pathway which allows our cells to make use of glucose. Uh, Glycolysis pathway. We need energy. Cancer patients, of course, need energy. Sadly, most of the energy is being diverted to sustain cancer cells. But then it is a trade-off uh, what you want to achieve. So many of these interventions do work before the cancer actually, the diagnosis is made for cancer, as we discussed in the very first lecture. There are, of course, possibilities to decrease the chances of having cancer. About 70% of them are caused by our lifestyle. Hope that answers the question. Okay, so let's resume this session again. No problem at all. Okay, you saw this. What is happening here? The cell is a cancer cell is trying to make some adaptations, and these adaptations simply mean that they don't rely on any metabolic pathway that requires oxygen. They learn, of course, to live with aerobic glycolysis, which gives them many benefits, as we saw. I know this is an energetically very bad process, but then there are lots of advantages of using this process. So what can also happen theoretically, what you can do in laboratory experiments, we know that if you cut down glucose, like Sandeep, when you were asking about glucose, what could also happen anytime this is done, glycolysis is inhibited, we know that the cells will try to adjust. And the way they do it is by enhancing the level of these receptors, the transporters, sorry, the glucose transporters. If you have more transporters, these cells will be able to attract more glucose. When you stop glycolysis, these transporters will go in number in order to try and compensate for the low levels of glucose. So one of the things that happens is this compound called glutar. This can be used to block these glucose transporter proteins. So the cells will have an imminent loss in the supply of glucose molecules to survive through glycolysis, and that will shut down their biosynthesis again. They will have low lactate, so they may become a very good candidate for attack by the immune system. But also, if you block this, what can happen is glutamine can be used as an energy source. And how that happens is that glutamine is converted into glutamate. Then glutamate can enter the TCA cycle via the alpha ketoglutarate entry point. So you can see in individuals who will have IDH mutations, they will have a deficiency of alpha ketoglutarate. But if you take a look at cells in culture which have deficiency of alpha ketoglutarate through IDH mutation, and you would expect to see alpha ketoglutarate is less, you may not find it. Why? Because it may be being supplied through the glutamine converting into glutamate and then entering the TCA cycle. So there are agents like glutaminase, this inhibitor CB839. So what can be done is you can block the glucose uptake by the cells using drugs like glutar, and simultaneously at the same time you can also block glutaminase through drugs, and that will have a very bad synergistic effect in terms of shutting down the metabolic pathways in cancer cells. You may ask, would it have any side effects? Well, on normal cells, yes, there will be some collateral damage. It may be that patients may not actually survive this kind of therapy and may experience some severe adverse effects, something that need to be checked. So have a look here what is happening in this example. This is lung cancer, and yesterday I was showing you one of the most prominent subclass of lung cancer. They have mutations in EGFR. We discussed this in some detail yesterday and day before. So this mutation in epidermal growth factor receptor, what happens here? These are very important in giving a signal transduction pathway green light to carry out this glycolytic pathway, which is important for lactate synthesis, as we know. It's also important for the synthesis of biomolecules, so anabolic reactions. Uh, what can be done again, if you take this labeled glutamine, you can actually follow the fate of glutamine, where this is going. You can also use uh, uh, the fluoro FDG, which is glucose, labeled glucose, and then see where this is going. You would notice, if you're able to follow these radioactive ligands, that many of them actually end up, the carbons end up in lots of biosynthesis pathways. So if you block EGFR using drugs like, this is a tyrosine kinase inhibitor, and lotinib, you can block this, and simultaneously you can use this glutaminase inhibitor to block this pathway. And you would see that this will actually have very pivotal synergistic effects on the growth of cancer cells. The problem again is, if you do any intervention like this, they work very well in cell culture dishes. Cells don't cry. You can of course look for markers like necrotic markers, DNA damage, and so on. But such therapies are something that may work initially very well in cell culture in vitro conditions, but as you progress through preclinical mouse models, these therapy may altogether fail. And that is why it is quite challenging to treat cancer which is sitting in between our own cells. You can see the situation as someone having a flu or a bacterial infection. They have different pathways, and therefore you don't see very many side effects for many antibiotics. At least this is what we know so far. But for patients undergoing cancer therapy, which is again sort of a parasite like a bacteria, there is not very much we can do as much for as for a bacterial infection. And the reason is, this is the product of our own cells. These are eukaryotic cells of our own body, which have now stopped following the norms, the regular norms of life, and become actually rebellious. That's why it is very difficult to treat cancer. So now we will change gears, but please don't forget, we are discussing these pathways with regards to cancer cells, their ability to try and adapt to low oxygen environment. And this will come again. This is actually very pertinent, sorry, to the new lectures. This one that I'm showing here on my screen. So what you're seeing here, one of the advantages which I did not tell you so far, is that the cancer cells, because of the altered metabolism, what am I meaning about with altered metabolism is none other than the requirement for oxygen is decreased. This allows cells to actually drive angiogenesis, and you can see here in some of these examples, the cancer cells, they're sitting here in the normal cells. These are tumor cells here. You can also see some cells undergoing angiogenesis. Looks like colorectal cancer. We'll have a look at this figure in the legend. So it looks like these cells are undergoing angiogenesis, making these new blood vessels supply, in order, of course, to make them more gain more nutrition, but also at the same time to help them spread as single cells to distant part of the body. Why this is important? Because as I mentioned, benign tumors, they they remain very, very small. They don't really metastasize. They are not cancer. They don't have very many changes to their DNA. Malignant tumors, they're growing much faster. You can see these tumors. This is inches. Have a look at this. This is almost 2 inches, this tumor. Really a big tumor. Inside this here, they must be experiencing a lot of deficiency of oxygen because it's a solid tumor mass, um, sometimes calcified, very hard to gain access to. And you can see what happens here is they experience lack of oxygen. But this lack of oxygen, as I discussed previously, has some advantages. But one of the most important advantage is that it allows these cells to form new blood vessels. So anytime any cells will experience this lack of oxygen, cancer cells specifically will start to form new blood vessels. And one of the ways in which they would do it is by making enhancing the level of a protein. We call this protein vascular endothelial growth factor. I'm sure you all know about vascular endothelial growth factor. And we need vascular endothelial growth factor for our growth. When we are growing, our muscle system, our blood circulation system, blood vessels, they need to be elongated, they need to become larger. And we need, in injuries also, we need vascular endothelial growth factor in order to repair the injuries as well. So what is known is that the production of vascular endothelial growth factor is, which is important for this to happen, new blood vessels to form. The vascular endothelial growth factor protein is formed more by cells when they experience hypoxia. So when the cells are experiencing hypoxia, I discussed you previously, they're relying, they're learning to rely on using glycolysis. They're trying to stop TCA cycle. They're trying to stop oxidative phosphorylation, which will kill them through the generation of reactive oxygen species, uh, through autophagy maybe. So they are trying to block those pathways, but also through the lack of oxygen, they are triggering the expression of vascular endothelial growth factor protein. The conditions of low oxygen, we call them hypoxic conditions. There are agents which have been used. There are drugs, experimental drugs, which have been used as inhibitors of vascular endothelial factor. If you block vascular endothelial growth factor, the cancer might just remain at one place, may not spread at all. If it does not spread, it's pretty easy for most tissues for doctors to remove them, right? But the problem with cancer is that it spreads, it metastasizes, it invades in the nearby organs. It spreads through metastasis to distant organs. That's the biggest problem, right? So vascular endothelial growth factor is something that is very important in driving synthesis of new blood vessels, a process we know as angiogenesis. So let's see what happens when our body, and I'm not talking about cancer cells, I'm talking about our body experiencing hypoxia. We have less oxygen in our body. So one of the causes of having less oxygen in the body will be what brings oxygen to the lungs in the first place. Um, what happens to the oxygen that is taken to tissues, how it is taken to tissues through the RBC. We know that. What happens is that there is an important hormone that regulates the formation of RBCs. We call this hormone erythropoietin. Now, erythropoietin is a type of growth hormone, which is again a protein. It is made exclusively by the cells. You see these cells in the cortex, the outer cells. These are the cells which are making the hormone erythropoietin. Now, what happens is there are patients in which As a result of chronic kidney disease, you have patients who have compromised kidney function. Uh, One of the bad things happen to them is low oxygen levels, low RBC count, and that is a result of destruction of kidney. If kidneys are damaged, they will not be able to produce sufficient erythropoietin. No erythropoietin means no RBCs simply, and less oxygen. Brain cannot survive without oxygen. It needs certain level of oxygen to survive. We all know that. So why am I telling you this? I'm just telling you how our body copes under physiological conditions when it experiences hypoxia or low levels of oxygen. And this is also relevant to cancer cells because they are, after all, product of our own body. So what happens? Have a look under normal conditions when oxygen is not deficient. There is plenty of oxygen. That only the specific cells in this region, cortex, they are the ones in the kidney who produce erythropoietin. You have less oxygen, RBC go down, of course, the expression goes up. What happens if someone is experiencing extensive hypoxia or individuals going on high altitude, for example, where oxygen is less, or individuals, of course, in cancer, as a result of hypoxia, the system, the body will try to counter the effects of hypoxia. In normal conditions, in normal individuals, red dots, not only the cortex cells, but also these medullary regions of kidney, they start to produce enormous amount of erythropoietin, as much as over 100 times amount of erythropoietin compared to these conditions. And that erythropoietin is going to send a stimulus to the bone marrow to make more erythrocytes or RBC. More erythrocytes will try to capture more oxygen and try to settle the condition so that the person returns to normoxia. I hope that makes sense. So not only human beings experience hypoxia, as I mentioned, cancer cells do as well. And I'll show you a pathway which is regulated by a transcription factor. And what this pathway does, in fact, do is to drive the expression of certain genes to help those cells survive the effect of hypoxia. Like these cells, what is happening here? There are cells which are making erythropoietin. But what is that pathway which is allowing them to make this erythropoietin protein, which is a hormone that triggers the synthesis of RBC? So you can imagine this pathway. The kidneys, what happens when they start to make and secrete erythropoietin, you can have more RBCs. But this is in order for kidneys to do that, they need to experience low oxygen. When the oxygen concentration decreases in conditions like anemia, most people will have their kidneys, if they're intact, if a person does not have a chronic kidney disease, usually kidneys will be able to enhance the level of RBC production through the synthesis of erythropoietin. The question is, what is making this erythropoietin? What is the proteins upstream of erythropoietin? What is the signaling pathway that actually does that? Have a look here at this pathway. So again, what you're seeing is a cartoon. And in this cartoon, you're seeing here a cell, a eukaryotic cell, and the nucleus. In the nucleus, there is DNA. Well, we saw that yesterday with the example of P53. We saw that yesterday, day before yesterday, with P53, and with yesterday with the example of androgen receptor. Look what is happening here. There is, like androgen receptor, there is a transcription factor, and this transcription factor is called HIF1 alpha. What happens under normal conditions? HIF1 alpha simply stands for Like androgen receptor, or A stands for AR stands for androgen receptor. HIF1 alpha stands for hypoxia-inducible factor. This is a protein made by cells when they experience hypoxia. And this is a protein which is also activated in kidney cells when they experience hypoxia. So what happens under normoxic conditions, meaning there is no shortage of oxygen at all, there is plenty of oxygen in the system. In these conditions, these oxygen molecules will bind to the HIF1 alpha through a covalent interaction. They don't bind just like that. They need an enzyme called PHD, which carries out this reaction, which carries out prolyl hydroxylation. So under normal conditions, in a cell, you will not find very much HIF1 alpha protein if you do a Western blot. Simple answer is HIF1 alpha is degraded. It is not there. Why? Because HIF1 alpha can be hydroxylated by prolyl hydroxylation, and then this hydroxylated HIF1 alpha becomes a substrate for VHL, which is a protein which, if it acts on certain proteins, any of its substrate, once it acts on those substrate proteins will be degraded. Their destination will be taking them to the abattoir and dying. Those cells will chop off through the proteasome those proteins which become the target of VHL. This is a simple E3 ubiquitin ligase reaction. So now, to tell you in another way, what is happening here, in normal conditions, HIF1 alpha function, which is a transcription factor, is not needed. When it is not needed, it is degraded. Of course, in normal conditions, you will have plenty of oxygen inside a cell, which will promote this reaction, right? The enzyme that carries out this prolyl hydroxylation is called PHD. That's the name written here. That's the enzyme causing prolyl hydroxylation, and that will degrade this. Do you know the cofactor for prolyl hydroxylase enzyme? This enzyme? The cofactor for PHD is the same protein, is the same metabolite, which is an oncometabolite. Can someone tell me what is the oncometabolite we discussed, which is produced as a result of mutant isocitrate dehydrogenase pathway? The name was not alpha ketoglutarate, it was 2-ketoglutarate. So what is happening here? The same alpha ketoglutarate, the product of a wild type isocitrate dehydrogenase reaction, that same enzyme, but wild type enzyme, gives rise to alpha ketoglutarate. If you want, I can show you that reaction again. Have a look here. Isocitrate in TCA cycle is converted into alpha ketoglutarate through the normal IDH enzyme. But if you have mutation, this IDH enzyme mutated will convert isocitrate into 2-hydroxyglutarate. What is happening here? These cells with this mutant enzyme will experience shortage of alpha ketoglutarate being synthesized through the TCA cycle. We know that now. So the same alpha ketoglutarate is needed by the PHD enzyme to become active. Once PHD enzyme becomes active, what will happen? HIF will be degraded. There won't be any HIF. There will not be any synthesis of genes which are needed for cells to survive in low oxygen conditions. Does that make sense? I hope it's not complicated. So 2-hydroxyglutarate is something which is produced through the mutant reaction, but in normal conditions, the wild type isocitrate dehydrogenase is making alpha ketoglutarate. So what is happening here? This enzyme PHD requires alpha ketoglutarate to become active. And what is happening? The alpha ketoglutarate will not be there. Why? Because there is a mutation in isocitrate dehydrogenase. What will happen as a result? The HIF will not be degraded. The HIF will become active. So we know what will happen once HIF becomes active is that the HIF will not go through this pathway. Under hypoxic conditions, there is no oxygen anyway. This PHT enzyme is not active. The HIF will translocate into the nucleus. And remember, P53 was binding as a tetramer to the DNA. Androgen receptor was binding to the DNA as a dimer, as a homodimer, two same androgen receptor binding to the DNA, driving transcription. In this case, what is happening, HIF1 alpha pairs up with ARNT or HIF1 beta, as we also call it, binds with this second protein, heterodimerizes, bind to DNA. Now you will ask me, so what genes does it regulate? Well, one of the genes that it regulates is erythropoietin in kidneys. Once this HIF1 becomes active, it enhances the level of erythropoietin expression that leads to the formation of these RBCs. So this erythropoietin being formed in enormous amount is because of the HIF1 alpha becoming active, as is shown here. The other protein, which is very relevant to cancer cells, which HIF1 regulates, and there are many, many proteins, in fact. Um, I'm just giving you some very simple examples. The other protein, one of the most important for the cancer cells to spread themselves throughout the body, this protein is vascular endothelial growth factor. VEGF, vascular endothelial growth factor, is also under control of HIF1 regulation. So HIF1 alpha, as a transcription factor, once activated, can also increase the level of VEGF in cancer cells. You can see here in normal cells, HIF1 alpha is degraded by the proteasome pathway through this reaction that we have discussed, the hydroxylation of the proline residues. There are specific proline residues within HIF1 alpha which become hydroxylated through this reaction, proline hydroxylation reaction. So what is written here is simply what I discussed. So what happens here, in a sense, is you have this tumor-associated angiogenesis taking place in tumor cells experiencing hypoxia, in kidney cells experiencing hypoxia. The HIF1 alpha will selectively enhance the level of erythropoietin. That's what is needed in a normal individual. But in cancer cells, they're also experiencing hypoxia. And once they experience hypoxia, they secrete vascular endothelial growth factor molecule. And this vascular endothelial growth factor actually stimulates, it drives the blood supply into them, so that these cells, the cancer cells, which are sitting in a primary tissue, can now actually spread to distant organs. So what scientists are trying to understand, of course, they see this as an opportunity. The cancer cells, they have a very low levels of oxygen. They experience hypoxia, while normal cells have plenty of oxygen. So they are trying to find drugs, interventions, which will specifically target and kill the cells experiencing hypoxia, meaning the cancer cells. And have a look at this molecule. Sorry, is someone asking someone? Kavish? Okay, no worries. So you can see this molecule TPZ, which has been synthesized. What happens with this molecule? This molecule specifically becomes reduced in cancer cells or the cells which are experiencing hypoxia. And this reduction of this molecule is indeed required to make it active at killing cancer cells. So this is now actually, this is a prodrug. This is an active drug. And one of the things it does is generate these hydroxyradicals, which will kill tumor by destroying the DNA. So these hydroxyradicals are formed. This will be very important. So the drug, although given systemically, perhaps like a tablet, is only going to become active in cancer cells because they're experiencing extreme hypoxia. They're growing very fast. I'm talking specifically about cells which are solid tumor cancer, um, experiencing hypoxia and will form these radicals. In order to destroy tumors, normal cells may be spared, may not experience as detrimental side effects as the cancer cells will. So with this, I can summarize this topic that we are discussing. The bioenergetics alteration in cancer cells is actually sits at the heart of these cells, which is important for cells to survive in a low oxygen environment. You can see how many things a cell is trying to do. This is super important for cancer cells to be able to survive in low oxygen environment because nothing, as we know, the life in primitive times when the earth was near newly formed, the environment was reducing, right? That is what the scientists believe. Um, Later on, it changed to oxygen. But those conditions, if created now, we will all die, except, of course, some bacteria which can make use of those conditions is still found in Earth, on the Earth. But otherwise, oxygen is so much required. But these cancer cells are excellent engineers in making sure, in training themselves to make sure that they have right pathways. So changes in the central metabolic pathway, not relying on TCA cycle, not relying on oxidative phosphorylation, which can kill them, relying mostly on glycolysis, trying to block the TCA cycle, trying to activate the HIF. All of it is they're doing in order to survive the conditions of extreme low oxygen tension. And that is why this altered bioenergetics in cancer cells is so important. We saw this oncometabolite, one of them, this 2-ketoglutarate is very important, but one of the things it is cells are trying to do is to decrease the level of alpha ketoglutarate, because high level of alpha ketoglutarate will not let HIF become active. If HIF is not active, the cells will not be able to synthesize new blood vessels, and if they cannot do it, if they cannot increase the level of VEGF, they will not be able to move out of the primary organ, and they will not be able to spread. If cancer cells don't spread, they can be very easily treated, right? So with this, I will stop this session and I will switch on to the tutorial, some of the omics that we do in order to identify prostate cancer drivers. Dr. Aziz, shall we take a 5-minute break before moving into this, or do you want me to continue? Okay. If no one wants a break, then we will take a break anyway after 15 minutes.
Yeah. Can I have a question?
Yes, please, Dr. Aziz.
How are you?
I am very well, except my voice.
Okay. So I was just wondering about the reports where the use of VEGF inhibitors made the cancer more aggressive. So it is being considered as a double-edged sword, which can, if you cut down the supply by using the VEGF inhibitors, they find alternate ways to survive, and that is even more aggressive and disastrous.
You are absolutely right. So there's a group, I think Rakesh Jain's group in Harvard. They made those inhibitors, and that is what I think cancer laughs at us, because anything we throw, it always find alternate pathways to grow. So even with these inhibitors, I would imagine many of these inhibitors are orally active drugs, and the way they are metabolized in the body may be very different. Of course, there is a window of opportunity. So perhaps the reason could be that we are not able to we are only achieving suboptimal levels of the inhibition that may actually trigger other pathways becoming active, which will drive cancer, which were otherwise missing. So you're absolutely right. But to really pinpoint what is happening here, I don't have an answer. If you can shed more light, that would be great.
Uh, no, actually, that's a very genuine concern because even in prostate, there
Mhm.
is uh, standard guidelines to wait and watch for some time.
Uh, You're absolutely right. So if you have, so the way prostate cancer is staged is staging called histological staging or a Gleason scoring.
Yeah,
It was after the name of a scientist from Canada, Gleason. But those people who are put under the watch are the ones whose tumors are not poorly differentiated. If you have very high Gleason score, meaning your disease is very likely to turn really, really nasty. There is no differentiation left. It is highly poorly differentiated carcinoma. Those patients will not be put in wait and watch. So if you have, for example, Gleason 3, Gleason 4 disease, there will be different interventions. So either those patients will be put under active surveillance or watchful waiting, sorry. So there are two things that are done to patients. If the tumor is really intermediate, then they will be put under active surveillance. If they're really low volume tumors, you know, I should point out that many of these prostate cancer patients who they just give away donate their bodies to cancer research. We find cases of incidental diagnosis, meaning they had cancer, which didn't say anything. They died of
Mm.
causes other than cancer, didn't say anything to them. Um, so those are probably low volume diseases. But on the other hand, we have a marker called PSA,
Mhm.
prostate specific antigen. If the values of prostate cancer antigen, for example, is normally it is under 1 nanogram per ml of blood, but if you find that the initial diagnosis you have a value of 20 nanogram per ml, it is very likely that the cancer has started to spread. And that is a very smart test. These are the sort of tests that we lack currently. Very minimally invasive. You can just take out the blood. Nobody minds giving some blood, especially if it can save them from any harm caused by cancer. But again, those patients will have likely developed cancer, and the values goes in like thousands
Okay.
sometimes of this piece. So that would depend what you want to treat. I would suggest what Sandeepan was saying. Eat less, do intermittent fasting, and enhance your
Mhm.
lifespan when you are healthy.
Thank you. Thank you. I do that very often, 16 hours or more.
Monday, Thursdays. Yeah, I think we should all do, if possible, who are fit, all intermittent fasting is a good thing. Any other questions before I go? I can discuss still those points that I was discussing.
Yeah, please. Sorry to keep you.
No, no, that's
And
fine.
we'll
I think
reconvene
the whole point
in
is,
10 minutes.
yeah, 10 minutes is good. So, yeah, in 10 minutes. The rest of you, if you would like, you can just type your questions, please, or we can discuss your questions after 10 minutes. Thank you. I guess we can start now.
Yes, sir.
Great, thank you. I think there is a comment or a question from Sandeepan. I'll take before I start. So it says, seems a dead end for fighting against the resilience of cancer cells. As an aspiring cancer researcher, it feels overwhelming how to combat these challenges at times. Uh, how would you advise me to look forward to this journey to navigate the solution for cancer, in particular strategies and aspects in this field? Uh, that's what exactly what I'm going to discuss in this tutorial. Uh, Sandeepan, we and Dr. Aziz can join this conversation by all means. Uh, when we are talking about cancer, it's not something that happens overnight. It is a manifestation of a very long journey of cells, daughter cells having mutations, again, the same cells having some other mutations, making them fitter to carry out these journeys. So they are in a way, as Dr. Aziz excellently puts, they have many targets which are moving. So they are continuously changing flexibly what they rely on in order for them to grow and progress. Um, so there will always be a debate whether we should try or not. I think for economies like economies, the question is, are we able to give our population the therapies which are already available to the wider public through private healthcare systems? If we are not able to give them that, then you also have the fibromuscular part of the gland. So you have fibrotic tissue within the uh, prostate gland as well as the muscular part. Uh, you can also have expansion of these parts, and that is often the case when you see someone who says, "Oh, my prostate hasn't become enlarged." Most of the time, it is not cancer. What they're talking about is a condition called benign prostatic hyperplasia. Now, benign prostatic hyperplasia is not cancer. It can be treated very easily. One of the treatments includes giving drugs due to steroid, but also patients in which the whole gland can be removed. So, when the diagnosis is made at presentation, you will have more than half of the population where the cancer diagnosis is made when the cancer is localized. And this is what Dr. Aziz was telling. These patients may be put on active surveillance or watchful waiting. They may not need a surgery depending on the how extensive the tumor is. Is this just one core? Are there multiple cores? That will also help uh, make a decision on the clinical course for that particular patient. The good thing is if the cancer is identified, diagnosed at this stage, the treatment is super well. And you can see, at least in UK, US, 100% patients survive 5 years after the initial diagnosis, if the diagnosis was made at this stage. So one of the things, as we've been discussing, sorry about that. One of the things we are discussing, cancer tries to invade, and we know cancer invades into the nearby organs. Does it need to metastasize? Metastasis is a different thing. It needs the supply to the blood vessels, to the blood circulation system in order to metastasize to distant organs. Always remember, uh, invasion is local and metastasis is distant. So in this case, you can see this prostate cancer is trying to go into the rectum. So the distal part of the colon, the large intestine, is also trying to invade into the bladder as well. You can see. So this is invasion. This is called locally advanced disease, meaning it is advanced compared to this one. And you will find this in many cancers. You will have cancer, which is a primary cancer. Uh, Patients may have diagnosis of locally advanced disease, which a prostate cancer is a very good example of. And what is happening here is that you see that there is, although local, it has not spread, it has not metastasized, but it is still advanced, meaning it has left the capsule. So prostate gland, the cancer is still encapsulated in this stage. In this stage, what happens is this is the advanced stage of the disease. And what happens here, if the cancer is diagnosed at this stage, of course, the chances of survival are far, far lesser. As you can see, this is a stage 4 cancer, meaning, and this is true for many other cancers, stage 4 would simply mean that the cancer has metastasized to distant organs. So this is a particular topic of interest in my lab, where we study the advanced prostate cancer using a variety of models. We also generate our own models to study what happens in this particular subtype of prostate cancer, which is lethal if it is not treated. So the way the cancer is treated, localized cancer or this cancer here, is by surgery and radiotherapy. Doctors know this very well, but students may not. You might ask me, so if there are two therapies, which one the doctors will choose? Basically, what happens for localized prostate cancer, which requires removal, surgery will be preferred. Uh, So there are people who are frail, who are really, really old, and they cannot undergo an invasive procedure, in which what will happen in those cases, those patients may be offered and will be offered radiotherapy. And this radiotherapy will destroy the tumor. Uh, with surgery, mostly it is radical prostatectomy, meaning the entire gland will be removed. The prostate cancer, which has gone into local invasion or metastasis, it will be treated by androgen deprivation therapy. Now, I was telling you yesterday we work on a transcription factor called the androgen receptor. And basically, the goal of this therapy, androgen deprivation therapy, is to block the activity of androgen receptor. I'll again show you that slide on androgen receptor. Again, so what is happening here? We have drugs like enzalutamide, which directly block androgen receptor. And we give patients therapy called androgen deprivation therapy. The intention is same. You're trying to block the activity of a major transcription factor, which drives prostate cancer. So this therapy is commonly known as castration. Castration meaning cutting down the supply of androgens. This can be achieved chemically tissues in the bones, and in doing so, they could actually dampen the tremendously the quality of life of a patient who has the advanced disease. And you can see the androgen receptor levels are going up. Like I was showing you the examples, have a look what happens when patients are being treated with androgen deprivation therapy, and what happens when androgen receptor becomes active in a cell. So this is what you're seeing is DNA inside the nucleus. This is a nuclear pore in a cartoon. This is androgen receptor, and it is bound by chaperone. And you can see there are at least 3 different domains. I don't want to overcomplicate, but these 3 domains are very important for androgen receptor to drive transcription. I told you day before yesterday that all transcription factors will need to interact with the DNA, and therefore they have something called the DNA binding domain. They may have different structures, but functionally their role is to allow the protein in which they're part of to take this protein to sit and bind to the DNA. But they have 2 other domains. One of them is called amino terminus domain, the N-terminus part of the protein, and they also have the ligand binding domain, which is the C-terminus part of the protein. In this cartoon, what you can see is that the androgen receptor is bound by chaperones. We know when androgen receptor is in a cytosol, it binds to chaperones, which stabilize the androgen receptor. We also call them heat shock proteins, the chaperones. Now, yesterday we were talking about a database of protein-protein interaction. That database that we discussed was called a string, if you remember. And we looked at what are the top binding partners of your favorite protein. We inputted androgen receptor and one of the proteins on top that has a score of 0.999, if you remember. That protein was none other than a chaperone. It was a heat shock protein. So androgen receptor binds to the chaperone when it is cytosolic. It is not active at the moment. So what is going to happen is that when the steroid hormone comes, it binds to the ligand binding domain, as is shown here. And the binding of the steroid hormone, androgens, testosterone, will cause this. They will bind to the protein. It will cause the receptor to change its structure so that chaperones can no longer bind to it. And what is now going to happen is that androgen receptor will be able to travel into the nucleus, binds as a dimer, which is not shown by the way here, and binding, it will also bind to some factors which we call androgen receptor interacting proteins. And what will happen, these are only few proteins shown. I was studying yesterday, we have identified over 1,000 proteins that bind to androgen receptor through our proteomic screen. So what is happening? You would ask me, hang on a minute. If androgen receptor is driving transcription, what is the role of RNA polymerase? Well, transcription factors actually create a platform for the transcription factor. RNA polymerase is a transcription factor. These factors, androgen receptor, for example, create an environment, create the conditions, create that very platform which we need for the RNA polymerase to bind to the DNA. And then RNA polymerase can drive transcription of target mRNA that they will want to target. One of the mRNAs which RNA polymerase regulates in this context codes for a gene, a protein called PSA, prostate specific antigen, which I was just mentioning a minute ago. So anything that you see in a patient blood PSA, the level of PSA reflects the level of androgen receptor activity. If you have high androgen receptor activity, you will have high levels of PSA. So what can you do in order to block this pathway, androgen receptor pathway, is to block the AR activation by the ligand. So you have these ligands binding to the androgen receptor. We have drugs. One of the drugs is called enzalutamide. And what you can do with these drugs when they're given as capsules, they block this ligand, the hormone, from binding to androgen receptor. And if these ligands cannot bind to androgen receptor, it comes back into the cytosol. It's not active anymore. And that is what is done using drugs like enzalutamide. This pathway becomes active. I hope this is crystal clear. One of the ways also that you can block androgen receptor activity is by blocking these proteins that bind to androgen receptor. If you can block these proteins that bind to androgen receptor in order to stabilize androgen receptor, you can actually decrease the activity of the androgen receptor. So either you can make new drugs that will block, that will compete with the ligand, that will compete with the androgen to bind to androgen receptor and activate it, or You can actually inhibit proteins that are part of the androgen receptor transcriptional complex. The major challenge is, first of all, is to identify which proteins bind to it. Unless you know which proteins bind to androgen receptor, you cannot think about decreasing the level of androgen receptor via interventions that target these proteins. I hope that is clear. So the proteins that bind to the androgen receptor, we call them androgen receptor associated factors, or ARFs, or sometimes we call them ARIPs, androgen receptor interacting proteins. That's one thing. Now, my laboratory works on both of these aspects. What we are trying to do in the laboratory is trying to develop novel drugs, novel AR inhibitors using approach one that will block the androgen receptor activity in tumors. The second thing that we are doing in the lab is to perturb the transcriptional complex of androgen receptor. If we make androgen receptor unable to interact with the DNA, it is not going to be able to drive transcription, is it? And as a result, the androgen receptor will become inactive. So these are the two approaches that my laboratory takes in the lab. To tell you more about in more detail, these are the three domains of androgen receptor, and you can check with your favorite protein of interest as well. If you are talking about the estrogen receptor that drives specific subtypes of breast cancer, essentially you will see these three domains because both androgen receptor as well as the estrogen receptor are part of the same superfamily of nuclear receptors, nuclear hormone receptors, as we call them. So, in the androgen receptor, we know what DNA binding domain is doing. The function of DNA binding domain is to allow the protein to bind to DNA. But in androgen receptor, we have the amino terminus domain, which is the major transcriptional driver. Without this, androgen receptor will not be able to synthesize mRNA of the target genes. And then there is a second driver in the ligand binding domain. So when we are studying prostate cancer with an intention to develop drugs to block androgen receptor activity, we are trying to target driver 1 or driver 2. Now have a look what happens with driver 2. This is the driver that I was talking to you about, the ligand binding domain, which binds to testosterone. Individuals taking testosterone for muscle building, that's a very bad news for them because this is something which can actually promote cancer diagnosis, taking excessive amounts of testosterone. And what happens? Binding of testosterone triggers the receptor activation. We discussed this on the previous slide. And once the receptor becomes active, it can actually cause prostate cancer. Not a good news. Well, we have very good drugs, thankfully, that can block the activation of the receptor called the bicalutamide, which is a generation 1, first generation anti-androgen. But we also have a newer drug called the enzalutamide. The functional analogy is the same. Both of them compete with androgens to bind to the ligand binding domain. This is how prostate cancer is being treated in the laboratories, along with androgen deprivation therapy is being treated in hospitals all across the globe. What we don't think about is targeting this driver, and there is a very good reason for not being able to target driver 1. The reason is that the driver 1, the structure, it does not really fold into proper 3-dimensional structures. It doesn't make those alpha helices or beta sheets. There is no defined structure. Unless you can define a structure of a protein, there is very little you can do in terms of thinking of blocking the function of protein. So you need a three-dimensional structure of a protein. Proteins don't look like they're shown on your screen. This is a simple, over simplified cartoon of androgen receptor domains showing you the functional domains. So coming back to this, there is nothing you can do. In the human proteome, substantial part of the proteome does not really fold into a proper structure. It gives them flexibility. Same is the case with, sadly, androgen receptor. The major driver of this disease, androgen receptor, is driven by driver 1 domain. And this domain is the major domain that drives the androgen receptor activity. And unfortunately, this domain is intrinsically disordered. It does not really fall into a proper structure, meaning it cannot be targeted by drugs. And in fact, we don't have any drugs at the moment which are used in the clinic, which will cure prostate cancer by targeting driver 1. Most of the drugs, in fact, 100% of the AR targeting drugs, which are used in the clinic for the treatment of prostate cancer, they target the LBD or the driver 2. Hope that makes sense. So what happens to the resistance? Now this will bring you back to the lecture 1 and lecture 2, the first day lecture and second day lecture. How does the resistance develop? There is no new mechanism as such. Where there is something new, I'll tell you. Have a look what happens. When prostate cancer patients whose tumors are being driven by androgen receptor, they're treated with first generation drugs, first generation LBD antagonist or LBD inhibitors. We see the receptor mutates. And the mutation are not happening at protein level, as you know, they're happening at the level of DNA, specifically within the exons. And here, what is happening? There are point mutations happening. And there are several point mutations that are known to happen within the ligand binding domain. As a result, what happens sometimes? The receptor in cancer cells becomes active despite being bound to the drug. So normally androgens binding to this receptor activated, anti-androgens or the AR inhibitors binding silence the function of the receptor. But because of these point mutations, the receptor is now becoming active by the very chemicals which were meant to stop this function. I hope that makes sense. So what would you call them, these mutations, as being? They are gain of function mutations. They've given them the androgen receptor ability to have an abnormal function that allows cancer to progress. Okay. We use these drugs. Hydroxyflutamide is one example. Cyproterano acetate, another example. Bicalutamide, another example. We use them for a very long time. 15 years ago, almost, we got new agents that can still target the ligand binding domain, but those new agents were active in androgen receptor mutant phenotype driven cancers. We call them second generation drugs. And these second generation molecules actually can inhibit cancer growth. They are saving lives. Few months to maybe a year, many patients actually get some relief from their cancer. Now you will ask me, so what is the problem? There are already second generation drugs. Well, as I said, and Dr. Aziz was just alluding to, there will always be resistance, sadly. And it did not take very long before the resistance emerged to second generation inhibitors. And you know what happened here? They had point mutations, but one of the most remarkable change that happened to them is that there was a loss in the exonic sequence, which codes for ligand binding domain. So now these cells in prostate cancer, which became resistant to second generation drugs, like enzalutamide. So doctors are giving enzalutamide. They're all very happy. We have a drug, enzalutamide, that works. Yes, it does work in patients for a good few months. The patient is healthy. They look all right. And then they're measuring their PSA from their blood. And the PSA is dropping. PSA is one of the genes which is regulated by androgen receptor. So a doctor, by just taking some blood, can make predictions that actually you are now in remission. Your cancer is gone. Your cancer is shrinking. On a scanners, it looks like the cancer volume is decreasing. The locally advanced disease, for example. But again, what happens? The PSA starts to increase upon subsequent blood test. Now, people don't understand what's happening. It turns out that the receptor has mutated again. And the receptor has mutated so that it does not really have the ligand binding domain anymore. But the major domain which makes the receptor active, the NTD, is still there, as you can see. So although the receptor has become shortened, any treatment with an intention to block ligand binding domain, including the androgen deprivation therapy, they're not going to work. Why? Because the receptor is now totally independent of the need of requiring an LBD. They don't have LBD. So if I show you some data taken from biopsies and androgen receptor level, expression level in IHC, established, have a look. These are the patients. These are matched patients. So pre-development of resistance and post-development of resistance to second generation inhibitors. And have a look what is happening. There is a tremendous increase in the expression of this small truncated variant of androgen receptor in patients who have developed castration-resistant prostate cancer. This truncated form you do not see in patients who are still sensitive to castration or have a castration-sensitive prostate cancer. So nature is always trying to improvise and finding ways. The nature is more steps ahead than we are in finding novel ways to promote in this context the cancer. You can see here there are point mutations in response to first generation, and one of the most prominent change that happens that leads to resistance to second generation inhibitors is the loss of ligand binding domain. But some of them will also have the loss of the some of them will have intact ligand binding domain despite having resistance to second generation inhibitors. They may have some new point mutations. That is also known. Recent data came from France which shows epidemiological genomics data showing that patients who become resistant to second generation drugs and drugs like DNA damaging drugs like docetaxel have a specific point mutations in the ligand binding domain of androgen receptor. But notice one thing. These cancers still require androgen receptor. They are still dependent on androgen receptor, either full length or the small truncated isoforms of the androgen receptor. So when this came in last few years, we started to look at, okay, so what is happening when you don't have the ligand binding domain? I didn't have to go much further than looking at my own PhD thesis, which I completed in October 2006 in Germany. So as a PhD student, I was, of course, I had left India for the very first time. I started my PhD in October 2003 to 2006. And there's, of course, a lot of work in the new lab, lots of pressure as well to perform. I had left two JRFs, both CSIR and ICMR. I had resigned basically, and there was no going back. So I started making these constructs of androgen receptor to see there are these domains, N-terminal domain, DNA binding domain, and ligand binding domain. What do they do? And I was studying proteins that bind to androgen receptor and seeing what is the impact of androgen receptor on androgen receptor of these proteins, those proteins bound to androgen receptor. So you see, I have been working in this area for a very long time. It actually enhance the productivity, maintaining the same research area. I did go here and there, but most of my focus throughout my research life has been on prostate cancer and largely on androgen receptor. So coming back to what I was showing you, these small truncations that are generated in a cells that lack the ligand binding domain, which are generated, produced as a result of cancer becoming resistant. I had done some experiments where I had, as a PhD student, where I removed the ligand binding domain. And I made these constructs, and I was looking at the effect of these constructs on AR dependent, the transcriptional activity of androgen receptor. So this is a simple reporter assay, which can allow you to evaluate how active the receptor is. So on y-axis, if you see a larger bar, it simply means that the activity is higher. And you can see here without androgens, androgen receptor is not active. You see very little activity. But as soon as in your experiment, you treat your cells with androgens, you see a 50-fold receptor induction. Activity is gone up. I hope that is clear. Sometimes if you remove parts of the NTD, you see that the receptor activity is gone, totally gone. So let's come to this point. I was making this, those mutations had never been found up until that point. But I removed the LBD thinking that because it is a driver, we will lose the activity of the receptor. But actually something opposite happened. As opposed to here, where there was a 50-fold increase, we saw a 3-fold increase in the receptor activity. So now the receptor has become super active as a result of losing this driver. So basically, LBD is indeed a driver, but there are constraints. When I say driver, it simply means the LBD needs to be engaged with androgens to be brought into the nucleus to carry out this function. But what has been happening is this molecule here, this protein, is able to come into the nucleus anyway. And look at what is happening. The receptor is now super active in presence of not only androgens. Have a look, even without androgens, the receptor is almost as active as it is in the presence of androgen, meaning the receptor has become constitutively active. I was showing you that battery inverter alternator, the switch the other day, which has on both sides. Basically, this is a classical example. You have, by removing the LBD, created a molecule that does not really require this androgen receptor truncation, does not require any androgen to bind to it and activate it. It's become active all the time. Now, pharmaceutical companies, drug industries, doctors can treat patients who have these androgen receptor variants. They can treat them with anything that targets the LBD. They're not going to see any efficacy, would they? No, because the target domain is missing. Why nature is doing this, we came to know a very, in 2006, but those eventually became clinically relevant after more than 10 years, and we started to see this as a result of new generation AR inhibitor drugs, that these truncations can be generated in patients actually. So we were able, in this case at least, we were lucky enough to predict this in before the resistance actually happened to this. So we modeled this resistance beforehand, as you can see through this work. So you can think this as a scenario where, in order to survive the attack from a predator, this lizard would shed their tail. And as you can see, this jumps and dance. And in doing so, the lizard is able to be fooled. They are able to trick the predator who will attack the tail. They again can grow tail. but they survive. And likely exactly something similar is happening here, where the androgen receptor, by removing one of the most important domain, survives any AR inhibitors which targets the LBD. So you can see here this receptor is now always active, even in the absence of androgen or in the presence of it. It always becomes active. Shall we take a 10 minutes break, Dr. Aziz? Okay, I think will stop. Okay, perfect. I'll see you soon. If you have any questions, please place them in the chat box. Great, we can start now. Thank you. So, welcome back, everyone. What we were discussing before we left is how this protein that is a major driver of prostate cancer, the androgen receptor, can become active despite the treatments intended to stop its function. And one of the ways it does so is by through mutations, which allow the androgen receptor to lose its ligand binding domain. And in doing so, the androgen receptor becomes super active, as you can see here. You can see here, this is an example I gave, and this is basically showing you the constitutive activation of the receptor. So it took many years afterwards to start defining what are the characteristic features of androgen receptor variants. These are smaller truncations of the androgen receptor I showed you. And these two papers come out. You can see here, one of them is in New England Journal of Medicine, which actually showed that having the presence of androgen receptor variant, one of the variants they study, which is one of the most common ones for the androgen receptor variant 7, leads to resistance to AR targeting drugs like enzalutamide and like the abiraterone. So this androgen receptor variant presents can serve as a biomarker for resistant for clinicians to make sure that the treatment that they're giving may not actually work intended to target the ligand binding domain. But then the good news is that there was another paper which came out from Memorial Sloan Kettering from Sawyer's lab, which showed that actually the androgen receptor variants do not function alone. Remember, I was telling you androgen receptor binds to DNA as a homodimer. It turned out that androgen receptor can also bind to DNA as a heterodimer. meaning androgen receptor full length and androgen receptor variants. The smaller form can actually bind together and then sit on the DNA to drive transcription and can lead to expression of genes. So what we ask is it possible that we can target both of them at the same time? Of course, androgen receptor, if you have a look at the structure, the androgen receptor full length has both driver domains intact, meaning the NTD, and the ligand binding domain. But the variant, the androgen receptor variant, the smaller, shorter truncation, does not have the major driver, which is a drug target. And sadly, targeting NTD has not been very successful to date. So why we are targeting androgen receptor signaling using AR inhibitors? Because as you see, this is showing you the drug use in a US prostate cancer drug market. And as you can see in these bars, these are different years, and you can see currently we are here. You can see most of the market is dominated by prostate cancer treatment is dominated by hormone therapy. Hormone therapy is nothing but the AR inhibitors that are used in the clinic for the treatment of prostate cancer. How big is the market? You can see over 3 billion US dollars. Some of this market is also covered by chemotherapy and immunotherapy and other target therapy. So, but most of the time you can see here the androgen receptor targeting strategy remains to be the favorite one for clinicians as well as for drug industries, and therefore there are efforts to develop better drugs. And you can see here the use of androgen receptor targeted therapy is only going to increase in the future. So what we are doing in my lab to support this targeting of androgen receptor novel agents, especially the variants and androgen receptor full length, is shown in this paper, where what we showed is that we can combine two drugs. One of them is enzalutamide, a drug that targets the ligand binding domain. And there is other drug, this is called EPI-001. This drug was in clinical trials, but since the clinical trials have stopped, they did not show any benefit. But it was a good tool compound to use in our experiments because of the fact that it can inhibit in culture dishes the NTD activity. This was from Nessa Pharmaceuticals. And we combine this drug with enzalutamide that targets the ligand binding domain using a linker. And what we could establish is that this drug, of course, this is a big molecule, will have lots of issues if given to humans or mice. But in cell culture dishes, we could see that this drug was much better at blocking the androgen receptor activity and therefore the growth of cells, because the prostate cancer cells in which we did this experiment, they were dependent on androgen receptor. So by doing this work, this research, we showed that actually gave the proof of concept that you can target the NTD and LBD together. And if you do that, then you will have a better killing and you can actually circumvent, you can stop the resistance from happening. So that was the initial work. Since then, and this work is published, as you can see, but since then we have developed better inhibitors. Please note that this is unpublished, and please make sure that this is not something you post on social media. So what you're seeing here is we use this compound enzalutamide, and we did structural activity relationships analysis and generated over 100 derivatives and identified this molecule. So this work is in the chemistry part is in collaboration with the Department of Chemistry, University of Cambridge. And you can see we made this drug duotamide. So If you Google this drug, you will not find anything at the moment. But basically, duotamide is a dual androgen receptor targeting derivative of enzalutamide, hence the name duotamide. And duotamide is, as you can see, it's a small molecule, and duotamide actually is capable, as you can see here, is able to bind. This is a FRET assay. Those of you who do biophysical analysis, biophysical assays, would know uh, what this assay is. But basically, what you're looking at is the competition, is the binding of duotamide to the ligand binding domain. And we are comparing it against other androgen receptor inhibitors, but also androgen receptor agonist or androgen receptor ligand, the androgen or R1881. And you can see in this experiment something very clearly. The androgen receptor ligand, the R1881, this binds to androgen receptor very, very quickly, and in doing so, it replaces a labeled ligand. And so androgen receptor in this all of these settings has a very strong fluorescent ligand bound to it that gives a very high polarization, uh, as shown here. But as you have anything that binds to androgen receptor which is not labeled, the label will start to go away. Who is best at it? The hormone, the androgen itself, the R1881. Now, if you compare this with duotamide or enzalutamide or bicalutamide, all three drugs are actually quite good in inhibiting the androgen receptor activity by binding to the ligand binding domain. What you can also notice is that this EPI drug, which targets, as I mentioned, the NTD, does not bind to the ligand binding domain. So we could establish that duotamide actually works like its parental drug. How? Because it can bind to the ligand binding domain. But also what we did is this biophysical assay called the SPR, or surface plasmon resonance. And what happens if you see the binding of the protein, the recombinant protein with the molecule, you would see a peak. And exactly this is the peak we saw when we used duotamide, but not in the negative control, not with the buffer alone. So we got an idea that actually duotamide binds to the androgen receptor NTD. So now, in addition to having a big molecule, which was a tool compound, which we used and proved that it can target both domains, we have a much smaller intact molecule which can target both the LBD and the NTD. So we use this in a reporter essay. I was showing you some reporter essays previously from my thesis. Have a look at this reporter essay. If we do a reporter essay to look at the transcriptional activation ability of androgen receptor, not full length this time, only the smaller truncation, the variant NTD-DBD. Notice the LBD is missing. What happens here if we use enzalutamide in increasing doses? You can see the doses are incremental. Do you see any decrease in the receptor activity? I don't. Meaning enzalutamide does not and cannot target this receptor. Why? Because the LBD is missing. Now we have drugs. Two of them were in clinical trials. This drug here, mesofinitin, from ESA Pharmaceuticals, and this drug here from Oncternal Therapeutics. Both are US-based companies targeting the androgen receptor N-terminal domain. Do you see any inhibition of the androgen receptor amino terminus domain or this smaller truncation variant? I don't see any. Then one of the postdocs in the lab did some experiments and showed that duotamide at increasing doses impressively inhibit the amino terminus domain of the androgen receptor, as shown here. So in cancer cells, which are driven, whose growth is driven by these smaller truncations, the androgen receptor is still active, and it is driving the inhibition. The duotamide is driving the inhibition of it. So because it is a target, any cell that harbors it depends on it. You block the androgen receptor activity, those cells, what should happen to them? They should die. And we tested that. Have a look here, what happens. These are some in vitro experiments and some tumor xenografts experiments we have done. Have a look what happens. These are 4 cell types, 1, 2, 3, 4. I don't want you to go into the details of the names, but have a look. This cell is driven by a full-length androgen receptor. This cell type has both full-length and the variant. These 2 cell types only have the variant. And then we tested 2 compounds, an LBD targeting drug enzalutamide, which is a clinical use standard of care for prostate cancer, or we tested duotamide. Do you see what happens here? With duotamide, you get a much better cell killing at the same dose compared to enzalutamide, which does not give that great inhibition of cell growth. And you see very similar things here in other cell types, which only express variant. So this is the sort of experiment that one would do to establish the efficacy of drugs. These are cell growth curves, by the way. What you are seeing in this tumor xenografts, that the tumors were established, shown in blue here. In this animal, you can see here, we treated it with a single dose of the drug. And you can see here what is happening. These cells now express both the full-length expressing cells as well as the androgen receptor truncation expressing cells. Both of them have heightened increased level of cleavage caspase 3, which is a biomarker for a cell death. So you're able, within this short window, as little as 24 hours, you're able to trigger apoptosis by duotamide in tumors, not the cells growing in a culture plate. So you will ask me, hang on a minute. This is the drug that you're developing, duotamide. These drugs have been in clinical trials, and you showed in your experiments they don't really work by inhibiting the shorter isoforms through this reporter assay. So what happened to these drugs? Have a look at their fate. So one of them, in the same family, the EPI 506 compound, they stopped because of the high pill burden. The patients were taking 9 grams of the drug, 500 milligram capsule, 18 capsules every day. This trial was terminated. You can check the trial currently available data through this identifier on clinicaltrials.gov website. And we in our hands, the reporter assay, we did not see any changes in the reporter activity whatsoever. Then there is this another drug that we saw, Oncternal Therapeutics drug. We did not see any reduction in the reporter activity or the receptor activity, transcriptional activity. What happened to that? Well, last year or in 2024, they announced that their drug, ONCT-534, the exact molecule I showed you, this one stopped to have any clinical benefit in treating patients who have castration-resistant prostate cancer. So both of them actually failed. So with duotamide, what we have found is a drug which can actually block both drivers, the driver LBD, which is a conventional driver, and the drugs used are the ones that are intended to target this driver. But we also have drugs like we also have duotamide, which can also block driver 1, which is the NTD. Any questions? Let's open floor. We'll take some questions and then we will move on. If you want, I understand. There is late in India, so if you want, we can crack on, continue. Your choice. Swati, let me know, Dr. Aziz, what you want me to do. You are the chair of the session. Samara is asking,
since it's not a
yes, Dr. Aziz.
conference, it's a course, so feel free. And I think it is 1 hour left, so we can go ahead. I think participants will be taking their break as and when needed. So whenever you need a break.
Okay, and we are recording this anyway. I don't need a break at the moment.
Okay, so
Thank you.
let's go ahead and
I'm more than
your
happy to do.
tutorial.
Thank
I
you.
think you had. You can start the tutorial.
So that is part of the tutorial as well. I'm showing some omics studies that we have done in the lab. As such, I will not be asking any questions formally. So
Mhm.
the exam will be tomorrow. So I do have access to that platform that was not working yesterday. So I might give it a try again as part of this.
Okay.
Uh, yeah. So what we are trying to do in the lab now is trying to understand what sort of proteome is regulated by this factor. Mm. The problem is when you use RNAi, because they're coded by the same genetic locus, the same gene, sometimes it's very difficult to get specific sequences which will allow you to get rid of the one form and not the other. So at least now in the lab, we have established a system. It is very recent, just last month. And what we can do, this is a Western blot experiment. And what you can see here, we are able to do is to get rid of, this is a cell line that expresses both forms, full length and the variant. The variant is shorter, so it is running faster on the gel. If you get rid of the full length androgen receptor, you can see the full length androgen receptor is gone. The smaller truncation is not changed at all. Or you can get rid of the smaller truncation using a specific siRNA, in which case this band is abolished and there is no effect whatsoever on the full length androgen receptor. And the GAPDH is used as a control here. So we have now these cells, we are doing this experiment in multiple replicates with an intention to carry out proteomics analysis with them to understand what are the targets underlying the androgen receptor, but also androgen receptor variants. Uh, good. So what we are also trying to do, I think I'll leave last half an hour for showing you that platform, and we can do some studies, uh, some exercises like the one we did yesterday. If you have your laptop with you, uh, so let me carry on for next few, I think 10-20 minutes, and then we can do that exercise as well. So what you're seeing here, I was talking to you about, is this paper that came. And this paper actually shows that there are mutations in individuals who become resistant to drug like enzalutamide, abiraterone. Both of them are second generation drug. So the cancer does not respond anymore. And they have found these new mutations. What they also found, and this is of course not surprising, that over 50% of the castration resistant prostate cancer have the loss of the TP53, so they cannot really make p53 tumor suppressor gene. And on top, what they found is that there is a new mutation which was not found previously, but now with new treatments, we have this point mutations which emerges in the androgen receptor ligand binding domain. This is the location of ligand binding domain. So overall, there are plenty of patients that express these variants. 50 to 60% patients express both the loss of function mutations in p53 and gain of function, perhaps gain of function. We didn't know up until this point, but what we did is to introduce both of these mutations by getting rid of p53, knocking it out, and by introducing layering on, knocking in this mutation L702H in androgen receptor in prostate cancer cells. Have a look at the results. This is a wild type cell, you can say, the one which was used for the creation. This is a parental cell. You can see here docetaxel is known to kill them, highly efficient. What happens when you get rid of p53 and on top of this you layer on, you include this point mutation, these cells, now the student in the lab calls them C42BX. You can see here, this is all unpublished by the way. You can see that these cells actually show resilience, they show resistance, and they don't die with incremental doses of docetaxel as the parental cell line died. So by having these mutations, actually the cells become resistant to docetaxel. And exactly this is the paper. This paper shows that these cells not only become resistant to enzalutamide and abiraterone, but also to docetaxel. So what we are able to do, what we have been able to do, is to show that these mutations actually emerge in patients. Which patients are these? These are the patients who become resistant to docetaxel. And basically, by doing so, we have created a new model of the disease. We have done some experiments in animals, and you can see here a very similar thing. These are preclinical experiments using the cell lines, parental or the new models. You can see here these xenografts, tumors grown on the back of mice subcutaneously. They respond to docetaxel and they die with incremental doses. But in the molecules in the AR where we have made those changes, they actually don't get killed by docetaxel. So docetaxel, by having these mutations, loses its efficacy. In other words, the cells become, by having these mutations, they become resistant to docetaxel. One of the things we are doing, I'll change gears and tell you, unless there are any questions on this. So what we are doing in the lab is trying to take medicinal chemistry approaches, developing these new molecules. What we are also trying to do is modeling resistance, and how we are doing this is using genome editing, as I've shown you the example. So we've made this cell line already, and these cells also show similar behavior towards enzalutamide. The AR-LBD targeting drug is not able to platform where you can identify protein-protein interactions. And one of the things we are doing is to identify what binds to androgen receptor. And I was showing you these 5, 6 proteins that, through the cartoon. What binds to androgen receptor? Well, what allows in the first place for anything to bind to androgen receptor? We have these proteins that bind to different domains of androgen receptor. We know some of them. Receptor is also undergoing some post-translational modifications. One most prominent one is phosphorylation. And as a result, there are lots of genes which are upregulated. We know some genes are also downregulated. Overall effect is growth. Androgen receptor activation. Samra was asking, what does it do in normal cells? It can reverse injury. Prostate, there is an injury in prostate gland. The androgen receptor signaling goes up. It can also cause inflammation, but in normal cells, it might actually help reverse the inflammation. And as we know, androgen receptor target genes can also be oncogenic. So if I show you this slide, this came from this Gunas Kandem paper from Sanger Institute in the last decade. Have a look at this slide. What they've done here is they've taken out different biopsies from the same patient. And each patient here, you can see they have a number. So this is showing you A10, A29, A21, A12. These are all different biopsies taken from patients, either from monoclonal seeding or polyclonal seeding, where one clone with some defined genetic changes has spread to different organs. So they isolated painstakingly these biopsies and did the sequencing, like what Dr. Aziz was mentioning to you with exome sequencing, RNA sequencing. And lo and behold, they found that in many of them, androgen receptor is either amplified, it has a gain of function mutation, it has these point mutations, but also this L702H mutation. They also have this T878A mutation, which my lab studies for the past more than 15 years now. So is this surprising to you? No. We know androgen receptor drives prostate cancer. What was surprising is actually a lot of these factors that bind to androgen receptor, they're also found to be amplified, mutated in those cancer clones. And again, this suggests that androgen receptor, and you have to appreciate not the androgen receptor alone, but many factors. Proteins don't function in isolation; they always work as a complex. There aren't very many proteins I can tell you on top of my head who only work alone. They need always coordinated activation, coordinated series of activation or repression. All the time, it needs other proteins that interact with your protein of interest. So you can certainly do this as uh, study and isolate proteins using proteomics, proteins that bind to your protein of interest by doing something shown on your screen in a minute. Dr. Aziz, is it fine that we continue for another few minutes and then do the small exercise, or is it uh, okay that I complete this today and then tomorrow do the tutorial? Tomorrow is a lecture, I think.
Yes,
Yes, tomorrow is
do
the
the
tutorial.
program
What would you
today.
like?
I mean, it's totally up to you. Whatever way is best for your.
Whatever. Is it possible to
Because we
do a
have time until
poll?
6 o'clock
I've not.
today, and
So
then
we
we
have
are.
50 minutes.
Yes.
Okay, that's good. Do you think we should do a poll, or shall we just continue? We don't have very many people today anyway.
Yes, you can do a poll, and you can even ask them if they are ready to speak.
Well, I just wonder if they're tired of listening to me. I'm not.
And I
Uh,
think this is what we cannot say for sure. I'm definitely getting feedback that yes, your lectures are pretty interesting.
Thank you. I would like to get some feedback. Yes, I haven't seen any. I'm not sure if there is any particular way to see it in Google Meet. Okay, if that's the case, I'm going to give you some examples now of how you can actually, for your own research, identify potential drivers. And uh, I think this is something which is still quite interesting. Have a look at this slide. And what you see here is nothing new. I was showing you some Xena heat maps yesterday. But we know androgens are the triggers for cell growth in prostate cancer. We know hence androgen receptor activation is seen in the prostate cancer patients. So long ago, as a scientist, not as an academic, we did some experiments and look at the kinases, because we know kinases are the workhorses of a cell. We wonder whether there are kinases regulated by androgen receptor. We found about 4 dozen kinases shown on this screen uh, as a heat map. And one of the top kinases was this choline kinase alpha. And this is a time course experiment. So you treat cells at time 0 and then take the RNA out at half an hour, 1 hour, up to 24 hours, and see what is happening to the gene expression of kinases. And we found many of them are actually not studied very well. So this is something that if you want to study in your research, by all means do so. The choline kinase alpha was quite interesting. It is a metabolic kinase, and we saw that the increase after almost 6 hours, it goes increase in expression. So first thing first, what we did is to get rid of choline kinase alpha using siRNA. And when we did that, we found that one of the pathways which was badly hit by getting rid of choline kinase alpha was protein folding. And that was perhaps not surprising. And the reason was there was a paper from Oxford which showed that there is choline kinase alpha even if it is not able to carry out its major function, which is to phosphorylate its substrate. If you introduce it in a cell, then it allows those cells to grow much faster, meaning it was oncogenic, despite not having the kinase activity. What we thought after this data came out that actually perhaps it is causing taking part in protein folding because these experiments were done in a prostate cancer cell. We thought perhaps choline kinase alpha activates and helps stabilize androgen receptor itself. You can see that we found when we use this is the data, original data that led to these pathway analysis data, which showed protein folding was badly hit by getting rid of choline kinase alpha, by the way. So we did test it, and you can see here we made some constructs, some cells in which choline kinase alpha can be, you can remove choline kinase alpha. There are two isoforms, and you can remove these by simply activating the promoter of the to activate the hairpin. Basically, this is particularly similar to doing sRNA. And what you can see here is that in both cases, in two different cell types, what you're seeing, doxycycline can lead to decrease in the levels of choline kinase alpha. So we also looked at the androgen receptor levels, and have a look. The androgen receptor levels are also decreased in both cases. Uh, Choline kinase alpha, we did a trial, a colleague of mine, in patients. We were able to access those tissue pre and post androgen receptor inhibiting therapy. And you can see here the level of choline kinase alpha mRNA goes down in patients who were treated with AR inhibitors. You can see here very nicely. So this is equivalent to probably this data, which shows that the androgens activate choline kinase alpha, while anti-androgens suppress it. So we know at least there is an involvement of androgen receptor signaling axis in this. And what you can appreciate here is that when these cells were injected in animals, you can see here the animals which were given the doxycycline, their tumors actually shrank. It did not grow as fast compared to these mice week by week, in which the choline kinase alpha gene was not switched off by such RNA. It was on like it is here. And that tells you that preclinically, choline kinase alpha is a good drug target because by blocking it, you can actually compromise, you can perturb the growth. And all of this lies, this ability is to control the protein expression of androgen receptor. We did some co-immunoprecipitation biochemical experiments and proved that choline kinase alpha and androgen receptor are part of the same protein complex. And therefore, we established that choline kinase alpha is regulating the protein stability of the androgen receptor. We did further biochemical experiments as shown here, and you can see without a doubt that if you take androgen receptor protein and challenge it, this is a recombinant protein, by the way, expressed in bacteria and isolated. If you take androgen receptor protein and have a look, I want you to, this is a complicated slide. I don't want to take a lot of time on this, but have a look what happens. Every time you treat cells with trypsin, this is trypsin here, what is happening in presence of trypsin, and this is in the absence of choline kinase alpha, you see that the protein is decreased. There, the protein amount is higher. If you take higher levels of trypsin, so not 10 nanogram, but 100 nanogram, and challenge the protein, incubate it with your protein recombinant, you can see here very little protein is left. Of course, we're using a lot of trypsin. But in this case, there is a protection which is not seen here, thanks to choline kinase alpha recombinant, which this protein was incubated with, that you see that the choline kinase alpha actually protects androgen receptors from being degraded by a protease. So basically, we were able to establish this. We published this paper. I was the first and corresponding author on this paper because I steered the study. We published this in JNCI. If you want to read that paper, you can ask me for a reference. Good. So, do we only study kinases, kinomes, and transcriptomes? No. The answer is you can actually study the proteome. Studying it is quite expensive in this country. I approached someone in New Delhi. I think they have a setup. Although the chemicals, the consumables cost are much higher in India because of uh, the technical assistance is slightly cheaper, or perhaps far cheaper. They turned out to be slightly cheaper in India, but I haven't done this with them. This was done in Cambridge. And you can see what we did in this experiment. Like we studied the transcript, like we studied the kinases, which can be upregulated or downregulated by androgen receptor. We also studied here the proteins which are regulated by AR inhibitors. And we found one protein which was called the PBK, mitotic kinase, which goes down not only by AR inhibitor drug, but also by getting rid of AR using siRNA. We found we did some staining using some human tissue with patient tumors or normal benign tissue, and you can see uh, these brown dots suggest that PBK is overexpressed in cancer uh, tissues compared to benign epithelia. You can see here. What we also established is that choline kinase alpha binds to the amino terminus domain of androgen receptor. This is a GST pull down assay, by the way. And it also binds to the ligand binding domain of the androgen receptor. Again, a kinase that can actually bind. So choline kinase alpha previously, this molecule, I haven't shown this, but it binds to the ligand binding domain, not to the amino terminus domain. But choline kinase, this PBK, we showed that actually it can bind to both ends of the receptor. The data is in the paper. And we've established this new platform where you can actually grow the biopsies, the prostate biopsies on these steel grids. So we have some steel grids which you can reuse. When fresh biopsies are taken out from the clinic, you can make the matrix that mimic the connective tissue, plate your tissues on this, and basically fill up to this point with the media and give your drug or your treatment to establish what is happening, to look for clinical markers, and have a look what happens when we give a PBK inhibitor. By the way, this inhibitor is not a clinical drug. It was a tool compound used preclinically. And I can tell you with my experience something. This came out of a cancer research paper. This molecule, PBK inhibitor, used, I think, in colorectal cancer. And they did very nice experiments. The paper, Cancer Research, is a very good journal, very reputable. And we use this exactly the way they've used it for colorectal cancer. Same mouse model, same dose, same method of delivery. You know what happened? The experiment failed. Mice became sick. And what happened is when we opened the mice to see what happened to them, it turned out using the same dose, same concentration, same vehicle, same mode of delivery, same days in a week, the drug did not reach the target organ. It precipitated in the gut. And we see some precipitation in the liver as well. Basically, it came out of the solution. And because the drug was, I think the name was TOPK032 or something, but because the drug was red in color, the powder, we could see it very clearly. So when you do research, please make sure you do your due diligence. There is very little you can do about published research. I mean, there are research forgeries, reports of people doing, research which is not really done, research which is fake, research which has been manipulated. That experiment did not work. We used the same method, same company, we ordered the chemicals. So at least the good thing was that at least it was soluble in vitro. We did not use very much drug. And you can see these effects you're seeing on Ki67, which is a marker for proliferation is through the treatment of this. It was soluble perfectly because we did not use very high levels. So I would suggest have a look. Make sure that the concentrations you're using, the papers you're reading, and the molecules that are there, are they going to be able to work or not in your models? So we were able to show that this drug is actually able to, let's see, the first author is a pathologist, and we were able to show in this paper that actually Ki67, the proliferative index, is decreased when PBK pathway is inhibited in such cancers. So I gave you an example, the choline kinase alpha. I gave you another example, PBK. Both of them are androgen receptor binding proteins. How many of them are there? We basically are clueless at the point until we did this proteomics study. I think what we have so far discovered is just a tip of an iceberg. Uh, how many we have? So how do you identify all of them? If not all of them, if you can identify through screens um, inhibitors, that would be a great thing to understand the mechanism of action. And these days, if you can go to some databases like PRIDE, so please note down PRIDE, proteomics dataset databases, you will find data from CHIP-MS experiment. CHIP-qPCR, you all know. CHIP-MS simply means that you can actually identify, you can chromatin immunoprecipitate, and instead of doing a Western blot like you do in CoIP, you run the samples into mass spec and identify through mass spec what your proteins are binding with. Exactly, this is something we did here using this technology. And this is CoIP part. I'm not going to go into the details of how you use antibodies to precipitate a protein. But what we next did is to run those proteins into a mass spec. When you're doing sequencing, you would do something different, especially if you're doing chip sequencing. You will, at the chip sequencing, you will degrade the protein and run the DNA into a sequencer to identify where your protein was binding. In this case, you're doing just reverse. You're degrading your DNA and running your proteins into a mass spec to see what proteins your protein is bound with. So that is something we did here and identified over 1100 proteins that androgen receptor binds with. So this is all unpublished. Please do not post this data. We found proteins that bind to androgen receptor in presence of the drug, and we found proteins that bind to androgen receptor which becomes downregulated or upregulated in presence of a drug. We have, how many can we study? Not very many. So if you go to the PRIDE website, you will find many of them in that data set that you can actually check. Many of them remain unexplored because it is a mammoth task. If you know that these proteins have been shown in a screen, there is no validation, there is no value. So one of the things you will need to do is to validate them. That is most important. So with this, I'm going to, I can in tomorrow's lecture I'll show you some of the examples, but for now I will try and access the GPA data set and show you what you can do with that GPA data set. So I'm going to stop sharing my screen and see if I can upload the GPA2 data set. Thank you. Okay, let me check. So that data set did not work yesterday as we were trying to access it. Okay, let's see if we can do that today. Could you please confirm that you are able to see my home page? Okay, that's great. This is an excellent website. So far, I've only uh, use GPA 2, but this is giving me now a new link to GPA 3. Essentially looks very similar. I've not used this before, but let's try this. So if you remember yesterday, what we were trying to do is to see which genes are co-expressed, meaning they follow the similar expression pattern. If gene A goes up, B goes up. If gene A goes down, B goes down. And we saw this with an example of 4 or 5 genes which were going up with the smaller concentration of glucose, and they were actually going far, far up. Their expression was increased by higher concentration of glucose. That's fine. How do you recognize, identify those genes, first of all? The second is I gave you an example using Xena browser, and the example was focused on androgen receptor high expression, high prostate cancer patients have very high levels of ARID1 as well. And the A1PG was just reversed. There aren't very many patients who have high androgen receptors, but they actually have very low levels of A1PG. I hope you remember that. If not, I can go back and show you. But let's crack on with this analysis. So what you can do in GPA, there are lots of things. I haven't seen this before. They're saying this is new, which is fantastic. Uh, I will try and work it out what it is. They used to have something. So this is expression analysis. And within expression analysis, what I want to do is correlation analysis. I want to see the level of correlation. I have a gene and I want to know what is the level of correlation of these 2 genes. Are they part of a similar pathway? Can we establish this? Do we know this? And this is something that you can do using this platform. And they give you an example here. This is RB2, which is part of the HER2 family, and TP53. Now you can access any of the data sets. There are example gene sets here. There are also the gene B that you want to look at, whether there is any correlation or not, and the tumor types. So you can use all of this information and ask a question. There is gene A, there is gene B. Is there any correlation? This is particularly similar to what we did yesterday. So let's say if I type in here androgen receptor and type here ARID1A and ask the same question we were looking at visually in Xena yesterday. We can look at here. Have a look at this PRAD data set. PRAD is nothing but prostate adenosis carcinoma. And what you can then do is, where is that gone? And you can click, can you see that plus button? You can hit it and it will bring it here. You can remove these 3 data sets from the analysis. You only need PRAD data set. Now you can ask this question, okay, we were seeing a strong correlation between the 2. It may not have been a top correlation. So you might ask this question, okay, is there any correlation? Can you generate a plot showing the coefficient, correlation coefficient in Pearson? You can change it to others as well. And you simply click run. You see what happens. Okay, it's not able to run it. It's saying for some reason gene set. Perhaps we didn't have that on. Let's try this again. Okay, gene set B is empty. So we will try and see added 1A. And again, click run. So it looks like this is not working at the moment. Let me go back to GPA 2, which I'm more confident in using. I've never This is new addition. Can you see GPA2? Can you confirm? GPA2 or GPA3? Okay, I think now you can see GPA2. Perfect. So if you go here and type in, you can actually do a lot of analysis again with this. If you type in GPA2, I didn't see that prompt in a GPA3, similar gene detection, asking for the genes which are top hit in terms of the expression, either same direction or opposite direction with your gene of interest. You can do similar gene detection analysis. And what you do here, simply put the name of your gene of interest. I'll do androgen receptor and see whether one of the top genes was ARID1A, which I was showing you through that Xena browser. ARID1A actually has a very good match. Patients who have very high level of androgen receptor have very high level of ARID1A as well. What about, let's see if ARID1A is one of those genes which will stand out in this analysis as well. So if I go here and type in the same, select the prostate adenocarcinoma data set and add, and then ask it to list all other genes. See if it can do that. Now it is doing something. You can see this is the analysis it is doing. It is trying to establish the transcript of those. This is again patient data, seeing patients who have high level of AR also have high level of this gene. It has already given me the results. So you can see here, this is the result of this analysis. And you can see many of them are actually not published data. We are working on some of them, not because we found them through this, but we later realized actually one of them is one that is shows the same expression pattern as does the androgen receptor. What do you see being the first one? SMC2. SMC2, it is telling me, has the highest score with androgen receptor, meaning SMC2, SMC2 and androgen receptor are part of a similar transcriptional pattern. So if AR goes up in some patients, they have high level of SMC2. If there is no AR in some patients, they also don't have high levels of SMC2, but rather low levels. So this is good. You can actually go and have a look at top 20, the top 100 if you want. But what you can also do is this will tell you whether these they may be part of the same transcription complex. Remember, this is transcriptome data. So it may be both transcripts are up. They may be up. Why? Because they're both making a lot of protein, and those proteins are part of a similar same transcriptional complex. Or maybe there is an upstream regulator which is regulating, which is enhancing the level of AR, but it is also enhancing the level of those genes, including SMC2. Meaning SMC2 may be a sister gene of AR in terms that it may be a target of an upstream regulator. That is good. But if you are asked to develop a plot, meaning where you can see the correlation with the use of a dot plot, for example, or a bar chart or something similar, what would you do? So in that case, what you would do is you will carry out this correlation analysis. And what you do here, please remember the name of this protein, SMC2A, and we can do something else. We can go the other way around and ask it to find the negative correlation. I think it doesn't look like an easy job to find the negative correlated gene. Okay, so we will stop at SMC2A and we will go and do the correlation analysis to generate those plots first and foremost. So if we go here, type in the gene name. The first gene is AR, no doubt. The second gene is SMC2A or SMC2. I think it was SMC2. And if we select the PIRA data set here, prostate adenocarcinoma, have a look what we get. And we want to see Pearson correlation plot. Let's see what does it plot. It's already plotted these. And you can see there is a super strong correlation, positive correlation There is a very good p-value. There is an this r square is very good, very high, 0.8 almost. And you can see there is, this is TPM, is nothing but transcript per million. It is telling you each dot is a patient. On that, I was telling you there were 560 patients or something. You can see patients who have very high, low levels of SMC2. This is log scale. They also have very low levels of the androgen receptor, or patients who have very high levels of SMC2. SMC2 also have very high levels of AR. So with this, you can actually start using your gene or your gene signatures, because you can also use your gene signatures. You can actually establish what are the genes which actually track with in terms of their expression with my gene of interest. This is something you can do using this platform. It also allows you something to do differential genes and other things, but also survival analysis. So you can actually do survival analysis of your own gene and generate Kaplan-Meier plots like we did yesterday. I showed you some Kaplan-Meier plots of breast cancer patients who have very high, or they have metastasis, they have levels of hormone receptor, they have presence or absence of HER2 receptor, for example. So if I go here and look at this transcriptomic data, ERBB2, what happens with ERBB2? We should be able to draw those plots using this platform. Uh, so I find it really useful to use this platform to establish firsthand what are the genes which track in terms of their expression with my gene of interest. And remember, take this data with a pinch of salt. This data is transcriptomic data. And I was telling you day before yesterday, in fact, yesterday, the transcriptomics and proteomics, so transcription and translation, they are not linear events. You might see a lot of transcript of a particular mRNA, but it is not translated to a similar degree, and does not have any protein or very little protein. So the end result is you will have to have a look at the protein levels instead of looking at transcript. But transcript actually is a very good starting point, very cheap to do. You don't need an antibody. You can get results much faster for a qPCR than you would for a Western blot analysis. So this is all good. Any questions so far? So if there aren't any, I will show you this SMC2 now. You can visualize it in the Xena platform. Remember, let's go back actually. If I am here in this platform and you take a look at two top genes, which shows similar gene detection, which means similar expression pattern. I'll show you in Xena as well how they would look. So androgen receptor, and I want to look at the levels of androgen receptor in prostate adenocarcinoma, uh, at them and ask them to list it. Please note the name of two top genes that you see. Which ones are these? So the first one I remember is SMC. Two. The second one is FAM, which seems like this is a solute transporter, FAM 120A. So let's go and have a look. FAM 120A and SMC2A. How do they look in Xena? So you can plot them. So once you get the data, you can generate those curves, the correlation curves. And let's see if we can access Xena browser. Yes. And if I say I want BRAD, prostate adenocarcinoma patients, yes, it will give me all those patients. And then I can type in the name of the first gene, the androgen receptor. That's what I want to see. And compare the expression with FAM120A and SMC2. So if I just go for basic analysis, gene expression, and type in the name of SMC2A, I should see exactly a mirror image of this, because that is the top gene with very high correlation, 0.79, which is not 0.9. That should have been the case if it was like a total mirror image, but still, let's see if you can get the same data SMC2. from this analysis, because the patient sample, remember, the patient sample in TCGA and the patient sample in GPA, they were all same. So this is just a different visualization technique. You want basic expression analysis, select data set, gene expression, basic gene expression, and ask it to plot it. You can see this is exactly the same. Uh, what was the other one? FAM 120A. And you can do gene expression, basic analysis, and done. Exactly the same. So this looks beautiful. You can see this is uh, exact copy of each other almost. So if you can zoom in, you will find patients who have high level of AR, have high level of SMC2 and FM 120. And that's why the program, the GP, identified these two as the top hits who follow the same expression pattern as does androgen receptor. And are you seeing these four arrows? If you are able to go there, you can actually bring SMC2 to the front and then measure the relative expression of AR and PAM2 with regards to SMC high patients or SMC low patients. Again, you see very similar picture. Patients who have high level of SMC2 also have high level of AR. Or you can bring the FAM20 to the front, and then all the data is relative to FAM120A, in which case you're seeing very similar expression patterns, patients with high levels of FAM2, very high levels of SMC2 and AR. Many of them, I can tell you, have not been explored. We don't know what do they do. So this might be a starting point, especially for those of you who are doing a BSc, a master's project. Certainly, we'll be able to produce some very high quality publication quality images using these platforms that we have discussed. So with this, I will stop and I will take questions. Are there any questions on this? If there aren't any questions, should we close the session, Swati? Yeah, happy to wait here. Okay, yeah, let's wait. So let's stop my share screen share anyway. And feel free to ask me any questions that you might have. Yes, while doing the analysis, do we have to set any parameters? So most of the things, I think they work by default. You can certainly change the axes and few other things. You can also, I should mention, both in GPA and in Xena, you can actually, for free of course, you can actually input your own data to do such analysis. Some parameters are set, you cannot change, but others, like for example, zooming in on particular subsets, you can certainly do that. It gives you all the functionality that you need in order to produce publication quality images. I hope that answers the questions. Any other question? Padmavati is asking, how do we know which is the best limit for such parameters? I think with regards to limits, for example, I was showing you an example and it draws the data, for example, in that correlation plot. It drew it in such a way that you have a good spread of the data across the board. So you had very, very low samples where there was very low levels of both SMC2A and androgen receptor. And then on the other side of the spectrum, you had very high levels, patients with high levels of both of them. And it actually set that parameter by itself. So you don't have to do very much with regards to this. It should tell you straight away. It should draw the chart in a way that this is, again, going to be easily interpretable. You don't have to set those parameters yourself. With regards to p-value, yes, I think there are options in this if you go, and that is true for larger data sets where you're not inputting just one single gene, you're inputting many other genes. We are developing some platforms where you can actually play around with the p-values if you want to. For example, if you get for your signature enormous number of like hundreds of genes with p-value less than 0.05, and you find it very hard to deal with, you can make the p-value more stringent. So for example, 0.01, for example, to get a shorter set. So that is something you can control. In some of these, as I mentioned, in others, it is not possible, like the one we are developing here. It is quite possible, but that is for a separate application. Okay, so Padmavati is asking a question. If you want, you can switch on your mic, but I understand if you don't want to. I analyze some lipidomics data using the Metabo analyst tool. But there are many parameters starting from different normalization methods. I think one of the best things to do would be to get help if you have your own data, to get some help from computational biologists. I should tell you that if you're using some of these platforms, omic platforms, to generate, for example, metabolomics data, best thing would be to get help from the platform itself. If you have some pipeline which has been established through some computational biology collaborations, they will be the best people to guide you. For example, when we are doing experiments, I'm a biologist, pure biologist, wet lab scientist. For many of the applications, I rely heavily on having the input from computational biologists because they are the best suited to tell me, okay, this is something the data is telling you, you need to use this type of a statistical analysis. So there is no 1 + 1 = 2 answer, sadly, for that, Padmavati. I've never used this tool, by the way, MetaboAnalyst tool. But if you want, you should certainly have a look at the tutorials. There must be lots of tutorials if this is a tool which is quite frequently used by other people.
The problem usually is that there is a lot of difficulties and user interface problems in freely available softwares. And the ones that are good, they are paid. I used to use Ingenuity from
Yeah.
Really, it was Ingenuity itself. But now I cannot afford it. And the bioconductor and other things, they are pretty difficult interfaces. So would you suggest something where there is a compromise?
I think, for example, pathway analysis, I certainly would say there are platforms like David Go, and this
David
is
is quite old. David has not been updated in years. So
and there is one more platform. No, I agree with you. For most of these applications, our own computational biologists, they use ORA, overrepresentation analysis, and they because they know programming, they can access these platforms, Dr. Aziz. So we don't have a license for IPA either. But yeah, I think if you're doing any high throughput analysis, science cannot be done these days because of its interdisciplinary nature. You can't really do it in isolation. So best to have some collaborators who have complementary skills and can actually help you facilitate such data analysis. I tried to learn many aspects of computational biology, but then I gave up. I thought I'm better off being a wet lab scientist. But I do have some idea of what I'm requesting. So when I have meetings with computational biologists, I tell them this is what I want to see. How does it look like in the data? Are there any sense that this data is making that we can take and translate into preclinical studies? And sometimes they works, other times they fail. Because computational biologists themselves don't have idea unless I tell them this is something I'm after. I don't have any idea of programming, so we complement each other very well, actually.
Thank you.
Thank you, everyone. I'll see you tomorrow.
So we'll reconvene tomorrow at 11, and then there's a lecture at 12, and then we have a break. I think all of you have the program. Please go through it. And tomorrow being the last day, please fill your feedback forms so that you can take the exam, and then you'll be given the certificates.
So Padmavati is asking Dr. Azhar what is going
Okay.
to be.
Tomorrow's exam would be based on Google Forms, and there'll be short one-mark questions, mostly MCQs, and they will be all from the topics that we have covered in these 5 days.
100 MCQs?
Yes, and we'll try to give them
1 hour?
as well. 1 hour time in 100, but they're doable in 1 hour. From our experience last time, yes, there was plenty of time left.
Okay.
All right.
Okay.
Thank you, everybody.
Take care.